non-disjunction, each nucleus should contain 23 chromosomes. You have to know that if it is a human cell, this is a human cell, that each nucleus must contain 23 chromosomes after meiosis. But if now non-disjunction occurs, it means that if the chromosomes fail to separate, if chromosomes fail to separate, that's what you call the non-disjunction during anaphase. If the chromosomes fail to separate, then it means that this number of 23 will not be brought about. So one nucleus. So now what happens in non-disjunction? Yes, okay, saying that you have a cell under metaphase I. And then now this cell has chromosomes. Remember, they are supposed to be aligned along the equator. You see, we're explaining the process of non-disjunction. They're supposed to be aligned along the equator. Now, in actual sense, in the normal cell, if it is normal, yes, we expect it to be like in anaphase. I expect it to have these chromosomes separating. You see, these chromosomes must separate. You see, they are separating. And then in telophase, I expect this cell to form two cells, whereby these two cells, they have two chromosomes. You understand? So if it is a normal cell, I expect it to be 46. Now this one is also 46. After, at the end of telophase I, I expect to be 23, and also this one 23. But if non-disjunction occurs, disjunction occurs, what happens? Metaphase, the chromosomes, yes, they will be fine like that. In metaphase, they will do what? They will be fine, yes. And then when they go to anaphase, now because we said it fails to separate during anaphase, yes, these ones are 46. If it's a human cell, then automatically is going to be, now what's going to happen? It's going to happen that the whole set, this whole set is going to go to one side. The spindle fiber are going to fail to contract from this other side. You understand? And then it pulls this one, also pulls this one, but this one is going to go on one side. You see, it's going to go onto one side. Now, this is going to result in a cell having, yes, 1, 2, and then 3. 1, 2, and then 3. And then the other one is going to have only one chromosome. You understand? So now, it means that this one is going to have an extra chromosome. This one is going to have an extra chromosome. Yes. And then this one is going to have is going to be less of one chromosome. So it's going to have an extra chromosome. This one is going to be less of one chromosome. So now, if this one is still 46, now this one is supposed to be 23, but it has an extra. Therefore, it's going to be 24. And then this one is supposed to be 23, but now it is short of one chromosome. It's supposed to have 2 chromosomes. Now it has 3. It has 1 extra. And then this one is supposed to have 2, but now it is having 1. It means that it's short of 1 chromosome. If we go back to a human cell, it's supposed to be 23, then it means that it's going to have 22. So now what happens? So because now it has three chromosomes, then in science we're going to call it trisome. We call it zome because it is a cell, the body, which describes a cell which has 3 chromosomes of the same kind on the 3 chromosomes of the what? Of the same kind. Then here is going to be, because this one is going to be mono. Because it's a cell, a body, then you're going to call it zome. So it's going to call it monosome. If this non-disjunction occurs on chromosome number 21, then we're going to call it trisome 21. We're going to call it trisome 21. Now, what happens? if this cell, which has 24 chromosomes, the trisome 21, trisome 21, if it fertilizes a normal gamete, for example, this is a sperm, and then fertilizes a normal gamete, which is the ovum, yes, ovum, then automatically what we're going to have is we're going to have a normal ovum. Remember, it has a normal cell, it's going to have 23. So now it's going to have 24 and 23. Then you're going to have 47. Remember, a normal person is supposed to have 46. Now, instead of 46, it's going to have 47. So it means that now this condition, because of a normal cell fertilizing the cell which has an extra chromosome on chromosome number 21 is going to result in what you call Down syndrome. It's going to result in what you call Down syndrome. That's how Down syndrome is being brought about. However, we can also have a situation because we say that this cell is having 22 instead of 23. So now if 22 fertilizes the one which is 23, yes, now it means that it's going to have 45 instead of 46. Still, this is also an abnormal individual. So a monosome, if it fertilizes a normal cell, it's going to become 45. And then a trisome, if fertilizes the normal cell, it's going to form 47. All this results in abnormality in an individual. So that's what we are trying to explain. So now, when we say that one nucleus of when non-disjunction occurs, as we say that one nucleus will contain 22 chromosomes. We have seen where do we get the 22 chromosomes? It fails to separate, and then one nucleus is going to have 22 chromosomes instead of 23, while the other ones will have 24 instead of 23. It means that this one is a monosome, and then this is a trisome. It is less of one chromosome, it has an extra of chromosome. When either of these resulting gametes fuses with the normal gamete is 23. So now, if this fertilizes this, 24 and 23 is going to be 47. 22 and 23 is going to be 45. That's why they're saying that result in 45 or 47 chromosome instead of 46, which is 23 plus 22, you get 45. We have explained that. And 23 and 24, you get 47 instead of what? 46. 23 plus 23. So this will lead to a child having what you call Down syndrome. And then the child will have 5 autosomes with 3 chromosome number 21. It means that chromosome number 21 is going to be 3 of them. That's why we call it trisome. That cell is called trisome because they have 3 chromosome number of the same kind instead of a normal pair and 1 pair of 6 chromosomes. So 6 chromosomes. So if there are 25 plus the 6 chromosome, which is X and Y or X and X, based on which gender is it, then it's going to become 47. And then the women over the age of 40 have a great chance of having children with Down syndrome. We're not saying that children, all the people who are 40 years and above, they're going to produce kids who are having Down syndrome complications. No, we are saying that the people who are 40 years and above, they have a great chance of having this mutation, have a great chance of having a baby with Down syndrome. I think you have ever seen people with the Down syndrome. You have ever seen them. So what are some of the characteristics of people having Down syndrome? Stocky body. The body is stocky. Flattened face with a small and broad nose, small skin. Yeah, you have ever seen these people. Let's look at a picture which has for Down syndrome here. You have ever seen this. So let's look at a picture. The folds in the inner corner of the eyes that appear to be slant upwards, and then a large tongue. These kids, they have large tongue. Actually, the tongue is always protruding out. Yes, with the small eyes. Actually, the eyes are tilted. Yes. And then you're saying that the broad hands, the hands are big and short. actually the fingers, they are big and short, they are broad, big and short. And then you're saying that wide space between the first and the second toe. If you check the, you may not see the kid because of the shoes. But if you get a chance, this kid to remove the shoes, you can see exactly what is happening. Yes. So now let's look at some of the difference between meiosis I and meiosis II. Here we said that we explain this, that if you want to know what the differences between meiosis I and meiosis II, just look at the phases. Prophase, what happens? In prophase, there is no crossing over, there is crossing over. There is in metaphase, chromosomes align along the equator randomly in homologous pair. Chromosome line along the equator singly. So, and then anaphase, homologous chromosomes separate to opposite poles, while the other one, chromatid separate. Yeah? Telophase, you form two cells. The other ones, you form 4 cells. Crossing over occurs in prophase. There is no crossing over. So you have to look at the phases. Okay, chromosome align along the equator in a homologous pair. You see the word homologous. In the homologous, this is the key point. In the homologous pair. Then the other side, the chromosomes align along the equator individually. This is the key point which differentiate them. Then they are saying that the chromosomes move to opposite poles. They move to opposite poles. They are not saying, they're saying chromosomes. It means that they were chromatids now, homologous chromosomes. So the chromosomes now, they move to opposite poles. What moves to opposite poles in terms of meiosis II is the chromatids. Chromatids move to opposite poles. Why do we call them daughter chromosomes? They have not yet developed to form real chromosomes. That's why we call them daughter chromosomes, but we prefer saying chromatids. Two cells form at the end. So we say that here you form 2 cells, while the other side you form 4 cells. You see the key point? Yes. And then our chromosomes number in meiosis I is halved. Chromosome, you have the halving here, you have the halving effect. You can't see because of the color. And then this side, the chromosome number is maintained. It means that you start with 23 and then you end up with 23. Yes. Then crossing over takes place. We say that there is crossing over, while the other side there is no crossing over. There is no crossing over. Basically, those are some of the differences between meiosis. What about the similarities? Do we have some similarities between meiosis and mitosis? Yes, obvious. There are some similarities. What are some of the similarities? Both processes involve division. In meiosis I, you have what's called the cytokinesis. Even in mitosis, you have what called cytokinesis, division of the nucleus. So in both cells, you have you have cell division. It means that new cells are formed. However, in meiosis, you form 4 cells, and then in mitosis, you form 2 cells, which are identical. Both process occurs in M phase. M phase is the phase whereby the cell starts to divide. The cycle is divided. Is divided into M phase. You have the G phase 1, the S phase, and then the G phase 2. All this is interphase. That's why we call that this is not a part of meiosis. And I can call this growth phase. Growth 1, synthesis, growth 2. And then this is where there is division, the real division which is taking place. Actually, it takes around 80 minutes for it to happen. While this one takes around the remaining from 24 hours. That's what it takes. Yes. All right. In both cell cycle, the stages are common. You have metaphase, prophase, anaphase, and telophase, both in meiosis and in mitosis. There is synthesis of DNA, both. You find the synthesis of DNA in interphase. You see here in the S phase, which you call synthesis. That's where you find the organelles and DNA are being synthesized from. Cytokinesis, division of the cytoplasm. Yes, cytokinesis occurs in the 2 cells. What about the differences between mitosis and meiosis? difference between mitosis and meiosis. Okay, here we say that it occurs is in the cell body, which you call the here we say that it somatic cells. Well, this one occurs in ovaries and testes, resulting in gametes. Production of genetically identical. These ones are identical. Well, these ones are genetically different. They are genetically different. This one results in 2 daughter cells, while this one results in 4 daughter cells. These cells must undergo maturation, interphase, so that they can mature, so that they form what you call real cells. We call them daughter cells because they are still young. Then you have 1 nuclear division. We don't have mitosis 1 and mitosis 2. No. So it means that the nucleus divides once. That's why we say 1 nuclear. Well, here we have 2 nuclear division. Here we have crossing over takes place in Prophase I. Well, here crossing over takes place. There is no crossing over in Prophase I. That's why they are always identical. While in meiosis, there is crossing over which takes place in Prophase I. Then here we say that it results in daughter cells being diploid. You start with the diploid. You start with the diploid 2N, you end with diploid 2N. While here you start with the diploid 2N, and then you end with haploid 1N. You see? Yes. Then here results in the somatic cells, where this one results in the gametes. So basically, those are the differences between meiosis and mitosis. So now let's look at. So I think our next class, let's it be reproduction. We're gonna look at the reproduction. I know that many people like this topic, and then even in my even in our new YouTube channel for Bright Doctor, we're gonna also discussing too much of reproduction. Yes, check the link below. Don't forget to subscribe and like, and also share to others so that they can also benefit from our classes. Thank you very much. See you again.
Please use the link below to get the notes, questions, and other videos. Don't forget to subscribe, like, and share to others for our daily new videos.
Yeah, welcome back, guys. Welcome back to our class. We are looking for distinctions and making sure that we pass. All of us, we must pass. We must go to the university. Nothing can refuse us from passing. This is meiosis. And the content creator today is M.Saidi as usual. We have exciting news. On our channel. To our learners who are using our channel, we'll be providing vouchers along the video. So it will be airtime. It can be 10 rand, it can be 20 rand, it can be 30 rand, 50 rand, 100 rand. So in each and every video we'll be releasing from now. Sometimes the first person to comment on our video, then we will take that person and then we'll give a voucher. Maybe it's a voucher maybe for KFC, a voucher for McDonald's. Yeah, as we grow, as we continue growing, you never know that maybe we will have to have big prizes for our channel. Number 2 is we have also opened another YouTube channel which is going to help us in we know that this channel is designated to students who are grade 7 to grade 12. We are still building it. We are adding more videos each and every day. We have more teachers who are going to be coming to be producing content. But now we have opened another channel which is going to be shooting to the university, and also at least know how you can make money when you're still a student. Yes, you can also ask questions. It can be questions for health, any question you feel like you wanna know, maybe, but not in the context of education only. There are some things which parents, people don't talk about. So this is the time This channel is going to be trying to answer those questions people have. You just drop a question or WhatsApp, and then we make a video concerning about the question you have asked. So guys, subscribe and then hit the notification bell, so that when you release any video in our channel, you just know that, or just click the link and then start watching the video, then get your airtime, get your prizes. Yeah, the vulture will be embedded in the video, and then the first person to spot that vulture, then will be able to use it. But that person who has got that vulture, please comment down that, yeah, I'm so and so, yeah, I've got the vulture. Yes, today we are going to be looking at what you call meiosis. I know meiosis is one of the difficult topics, very simple to take, to grasp, but when it comes to answering questions, people get a lot of problems concerning about this topic. Yet is very easy to understand, but it is very complex when it comes to answering questions concerning about it. So what is meiosis? So you can visit our website, www.preprepthundereduc.com, and then, or if you have any challenge question, then you can also WhatsApp on that number, that is M.Saidi. So I'll be able to try to answer your questions. Let's look at meiosis. Meiosis, we said that meiosis, human reproduce, other organisms reproduce, they multiply. What about cells? Cells, they have two kinds of reproduction they undergo. They have what called mitosis, and they have what called meiosis. Mitosis can occur in other body cells, but meiosis occurs only in the reproductive cells. Where did you come from? Actually, meiosis was the basis of you living. Without meiosis, you wouldn't have existed. So let's look at meiosis in detail. So it's a type of cell division that results in 4 daughter cells, each with have number of chromosomes to that of the parent. So it means that meiosis starts with the one cell. This cell divides, and then you form 4 daughter cells. These cells, they have the chromosome number to that of a parent. If you have started with the 4 chromosomes, it means that these cells, they will result in 2 chromosomes. That's why you're saying that half the chromosome number of that of the parent. If the the body cell is having 46 chromosomes, then the reproductive cells or the daughter cells which are resulting from that are going to be having 23. And then from the mother also 46, they are going to result into 23. So the process of reducing these cells into have the chromosome number, that process or that reproductive process is what we call meiosis. So what does meiosis result in? So this meiosis will result in the production of gametes. Gametes, these are the sperms and the ova, and when you're talking about the humans, and then plants, you're talking about spores, and the pollen grains, you can talk about the ovules. Those are some of the things which are as a result of meiosis. So in humans or in animals, it occurs in the ovary. See this colored word, in the ovary. So in humans or in animals, basically ovary are found in female, and then these ovary, they produce what you call the ova. So the reproductive cells in the females, we call them ova. If it's one, it's called the ovum. If there are many, we call them ova. And then in male, we call it's occurring in testes. And then in testes, what we call them spermatozoa. The cells which are being produced, we call them spermatozoa. So in female, we call them ovary. Those are the ova. Ovaries, the ovaries produce the ova, and then in the testes, they produce what we call the spermatozoa. What about in plants? In plants, occurs in the ovary still, but what it produces, ovary produce what we call the ovules. Meiosis occurs in the anthers, and then what it is being produced is what we call the pollen grain. So the question can come and say that other than the ovary in animals, which other place does meiosis occur? That is testes. And what does it produce? It produces what we call the spermatozoa. And then in plants, it occurs in the ovary. It produces what you call the ovules. Basically, that's what you needed to know about this first introduction of meiosis. You know, meiosis is basically the cell divides, and the most important thing to divide is the nucleus. So we need to know the nucleus to do a recall about the nucleus, because we saw the nucleus at the beginning of DNA and also saw the nucleus in the grade 10. So let's look at the nucleus, the centrosome, and then the cytoplasm. Nucleus contain the genetic material. What about cyto means a cell, and then plasm means a fluid, the fluid of the cell. So basically, this is what you are going to look at. Let's look at them in detail. So this is a cell. It has the nucleus. Outside, you have what called the cytoplasm. Then in the cytoplasm, you have what called the centrosome. Centrosome, why do we talk about the nucleus, centrosome, and the cytoplasm? Because the DNA, you'll find it there, and then it is the centrosome which produces the centrioles, and the centrioles produce spindle fibers which hold the chromosomes in position. That's why you talk about it. And then there is a time when this one disappears, and then you remain the content of the cytoplasm. So the nucleus disappears, and then remain only with the content of the cytoplasm. So basically, that is the nucleus, and then when it brings it in exam, it looks like this. So this is the nucleus. So basically, that is the nucleus, but the nucleus has a dense body which we call the nucleolus. Then outside the nucleus, you have the nuclear membrane, which has the extension which we call the endoplasmic reticulum. But because it has the ribosome, they look rough. Then we call them the rough endoplasmic reticulum. The rough endoplasmic reticulum. So basically, that's it. It has the nuclear pore, the holes where substances go through, most especially messenger RNA in this case, and other substances. Then you have what you call the nucleoplasm, the plasma of the nucleus, meaning that the fluid of the nucleus. The nuclear envelope, that's what you call the nuclear membrane. Then the ribosome, we say that the ribosome does sites for protein synthesis. Chromatin, this one is the one which condenses to form chromosomes. And then you have the nucleolus. Then you have the rough endoplasmic reticulum. So we have explained all that. So when you look at the nucleus, or when you look at the DNA, basically that's what you see. You have, when the chromatin network condense, you form what you call the chromosome. So the chromosomes, they condense. You see, they have condensed, and then after that you form the real structure of a chromosome. It looks like this. So that's the structure of a chromosome. Now let's look at the chromosome in detail. So we have seen where the chromosome come from. It cannot exist without DNA, with its specific genes. You see, it has specific genes. So let's look at the structure of a chromosome. Why do we need to look at chromosome? You resemble your father because of the chromosome, because of the genes you obtained from your parents. Your father, your mother. What about those who don't resemble the father and the mother, but they resemble their grannies? We shall see. Another question, what about why? So in this topic, we're gonna see why. Why do you resemble your neighbor? Why do you resemble when you go somewhere? When you go somewhere, someone can say that, hey, you resemble someone. Why do you resemble someone? Those are some of the questions we are going to answer, and this is the reason why we need the structure of the chromosome. So the structure of the chromosome. Chromosomes are thread-like structures present in the nucleus. They're thread-like. It means that they are like strings, thread-like structures found in the nucleus. That's why we talked about the nucleus, which carry genetic material. That's why you resemble your father. That's why you resemble your mother, which carry genetic material from one generation to the next generation. So it means that your kids will try to resemble you. Yes. Then you're saying that each chromosome is made up of the DNA. We saw this, that DNA genes are almost the same thing, but on a different scale. So these DNA or these genes are tightly coiled many times around a protein called histone. So the histones, we saw this when we are talking about protein synthesis, and also when you're talking about the structure of the DNA. Yes, so they coil around to form what called the histones that support the structure of the DNA. Because the genes cannot just exist there in air, they need to have a support. So the support we call them histones. So these are the support which make the DNA strand to stand or to be where it is, and so that the chromosome can be formed when this DNA condense. All right, this is the structure of chromosome. Before, remember, we talked about during DNA replication that when does it occur? We say that it occurs before the cell divides or before meiosis. So before the cell divide, the chromosome will look like this. Yes, they will look like this. Therefore, we call this one single-stranded chromosome, chromosome 1, chromosome 2. Sometimes we call them unreplicated chromosomes. They have not yet done DNA replication. But the moment DNA replication occurs, this chromosome is going to change from this format to this format. That's why we say that it doubles the chromosome number. Don't say that it increases the chromosome number. It increases the chromosome number. It increases the genetic material. From what to what? The best answer is double the chromosome number. Doubles the chromosome number. So that when it was like this, then it becomes like that. So this is the centromere, the center of the chromosome. We call it centromere. The two chromosomes which look alike, it means that they share the same structure. Yes, and the position of the genes on them, then we call them homologous chromosomes. Homologous chromosomes. So one chromatid from one chromosome and another chromatid from another chromosome, because they are homologous chromosomes. Therefore, because we are talking about chromatids of homologous chromosomes, then they are going to become homologous chromatids. Why? Because we say that they are coming from homologous chromosomes, but each part is a chromatid. Therefore, they become homologous chromatids. So sometimes, as I say, that this is called unreplicated chromosomes, and then we call these ones the replicated chromosomes, or what you call homologous chromosomes. So each of these has two names: unreplicated chromosomes or single-stranded chromosomes, and then these ones double-stranded chromosomes or replicated chromosomes. So the number of chromosomes in each cell is a characteristic of organisms. For example, humans, we have 46 chromosomes. You'll find out that Drosophila melanogaster has 8 chromosomes. You'll find out that the sheep has over 60 chromosomes. So each organism has a specific number of chromosomes it has. And you might find out that the biggest organism has fewer number of chromosomes. So that's how life, or that's how God is. So it doesn't matter whether you are big or small, but you might have your own uniqueness. We are saying that chromosomes which are single thread or strand become double when the chromatid joined by centromere. So the center of the chromosome, we call it centromere, as a result of DNA replication. So if DNA replication occurs, as we said, now the single-stranded chromosome are going to become double-stranded. So that's why you call it unreplicated chromosome or single-stranded chromosome. The moment DNA replication occurs, it becomes double-stranded chromosome or replicated chromosome. Yeah. So what are some of the difference between the haploid and diploid? Di means two. Ploide means you are describing a body. Yeah, so half means it is derived from the word half the chromosome number. If a human being is supposed to have 46 chromosomes, so when you are haploid, or when the structure is haploid, it has the half the chromosome number of that organism. For example, if an organism has 20, the haploid state of it is going to be 10. If the organism is having 32, the haploid number of that organism, or haploid state of that organism is going to be 16. So haploid means half the chromosome number, diploid means doubling, doubling. What does it mean? Where is the double there? It means that half from the father, half from the mother, and then they double it. When they double, then they form what called the diploid or diploid, the way you pronounce it. So haploid is the quantity of the cell or organism having single set of chromosomes. They have what called a single set of chromosomes. So if you have chromosome number from 1 to 2, from chromosome number from 1 to 10, these chromosome numbers, these chromosomes must be in pairs. So if you are haploid, chromosome number 1 to 10, they will be in single. They will be 1, 1, 1, 1. They won't be in pairs. So that's what you call the haploid. But if it is diploid, is when the cell or organism has a paired or two sets of chromosomes, paired or two sets of chromosomes, one from each parent. It means that one from the father, one from the mother. When they combine, because there are two organisms contributing this, then you form the word di, which is 2. And then ploidy means a body. Yes, it's a state which describes organism with two sets of chromosomes, one from the father and one from the mother. That's the meaning. So for sexual reproduction to take place, sexual, it means that it must include the gametes. It must include the gametes and it must include the sex. Reproduction to take place, yes, what happens? A haploid male gamete, that is, if we are talking about humans, we're talking about sperms, will fuse with a haploid. It means that it will fuse. When you say a haploid male gamete, half of the chromosome number from the male, the gamete will have half of the chromosome number from the male. If the male is 46, the gamete is going to have, or the sperm is going to have 23 chromosomes. Will fuse with the haploid number from the female. Also now the female is going to produce 23. When they fuse together, you form fertilization. So when they fuse together, that process is called fertilization. Now you form what you call a diploid, because half from the father, half from the mother, then because it's half from different people, then it becomes di, becomes 2. They fuse together. And then when we're talking about fusion or fertilization, we are not talking about sex, because the fusion or fertilization can take place without sex. Meaning that maybe they used a male just give out the sperms. So when you're talking about fertilization, we're talking about sex has already occurred, and then the sperms, they swim along the female reproductive system, and then fusion will take place. This fusion can even take place. Someone had sex at night, and then you're walking on the street, fusion is taking place. Imagine, did you know when was your fertilization took place? Fusion is taking place. You don't know. Even your parent doesn't know. Okay, those are the wonders of the science. All right. If you need more questions concerning about this reproduction, go and visit our YouTube channel, which you call Bright Doctor. Bright Doctor. I'll give you the link in the description below so that you can understand it better. All right. The result is a diploid zygote. The result is a diploid zygote. So when you have a haploid, you form a diploid or a diploid zygote. So it means that when the sperm fuses with the ovum, you form what you call a zygote. But this zygote has a diploid number of chromosomes. This body has a diploid number of chromosomes. It means half from the father, half from the mother. All right, haploid, half, which is indicated, haploid is indicated as 1N. And then diploid is also indicated as haploid from the mother, 1N. For example, haploid from the father, for example, is also 1N. So when you combine them, you form the diploid. which is whole. Yes. So it's the whole. Now half and half you get whole. Yes. Basically, half from the father, half from the mother, from the diploid. Di means two, meaning that contribution from one person and contribution from another person. So because this resultant body is having two contributions, then we call it a diploid. So it is indicated as 2n. We shall be using this language, so you have to know when someone uses this language, you must know what it means. So a sperm, ovum, and then you form a zygote. Zygote is where a fetus develops from. We shall see in the videos we're gonna be releasing soon how is the zygote form. You see the sperm swimming after sexual intercourse. You see the sperm swimming going to the ovum and fertilization takes place. For more detailed about this, visit what you call the Bright Doctor. Bright Doctor, just link below. Drop your question, then we shall give you the answers on that YouTube channel. But for purpose of passing exams, we don't go far away from this. But there we can describe as much detailed as you want. So sexual cells is either sperms and ova are haploid. So we call them haploid. They are 1n, 23 chromosomes in humans. So it means that other organisms have different chromosomes. So we have what called the somatic cells. So it means that we have two types of cells. We have what called the sex cells. Those are the gametes. I say that meiosis can only take place in these cells. We don't find meiosis in any other cells, only in the gametes. So you have the other body cells. Soma comes from the word soma, which means a body. Yes. So these are body cells. A diploid, which means that they are 2n. If they are 46, it means that the haploid is going to be 23. But the question when it comes, it does not bring only human. You can only talk about 46 if they have specified in the question that this is a human cell, because they can bring a cell with 10 chromosomes. They can bring a cell with 4 chromosomes. If they don't specify that this is a human, please don't use 46, because the question is not asking about humans. Then you have the 23 pair of chromosomes that result in the zygote are divided as follows. How? Number 22 pairs. These are the number of chromosomes we have in the body. 22 pairs. That is from pair number 1 to pair number 22. We call them orosomes. Orosomes. It means that how many chromosomes do we have? 44, because this is a pair. Pair number 1 up pair number 22. We call these ones orosomes. One pair of sex chromosome, that one pair, that is the 23rd pair, we call it sex chromosome, or scientifically we call them gonosomes. Scientifically we call it gonosomes. So this pair is divided into two. We have what called the XX, which is female, and then we have XY, which is male. So it means that females always, they will have XX, and males will always have XY. There is a question which is supposed to be posed here. Why, if females are XX and the males are XY, it means that it's the males to determine the sex, because female have X, also male has X, but female don't have Y, but male have Y. So it's the males to determine the sex of the human being, the sex of an individual. So males, you must stop saying that ladies are only giving you females, they're only giving you boys, they're only giving you girls. Say, I'm giving myself boys, I'm giving myself girls. It's you who is determining this. However, there are some other factors which also determine. It can be on the side of the female. For more details, visit the Bright Doctor. drop your question there, we will be able to answer those questions. If you drop the question there, then I'll be able to answer those questions. Humans have what we call the chromosomes. We say that from chromosome number 1 to chromosome number 22. These chromosomes are arranged in a specific order and they appear in a specific way. They are in pairs, and then they describe different characteristics in the organism. So let's look at the karyotype, the composition of the nucleus. Which kind of nucleus? Which kind of nucleus do you have? The chromosomes, the type. Karyo is derived from the nucleus and the type. So which type of the nucleus do humans have? Thus derive the name of karyotype. Humans cell are made up of 46 chromosomes. It means that our nucleus have 46 chromosomes. And then these 46 chromosomes are arranged in pairs. These chromosomes are arranged in pairs. It means that they cannot exist as singles. What does it mean if you're not married? All right. It means that they are arranged in pairs. Each pair is different from each other. Each pair is different from other. So these pairs are different. By how do they differ? How do they differ? They differ by shape, size, and genetic composition. So pair number 1 is totally different from pair number 2 by size, shape, and the genetic composition. It means that what pair number 1 determines is not the same thing what pair number 2 determines. For example, you'll find out that chromosome number 23, pair number 23 will determine the sex. You cannot find this in other chromosomes. So they vary in shape, size, and the genetic composition. From pair number 1 to pair number 22, number 1 to 22 are called autosomes. What about 23rd pair is called gonosomes, which has XX for female and the XY for male. X, you have to know this, X is longer and bigger over Y. Why? X, you have to know this. X is longer and bigger over Y. Question: why? Why is X longer and bigger? Right, doctor. We'll answer that. But on the purpose of this, X carries more genes compared to Y. So a karyotype, an example looks like this. You see that chromosome number, pair number 1, number 2, number 3, number 4, they vary in appearance, shape, and also they vary in the genetic composition. So from 1 up to 22, we call them we call them gonosomes. We call them autosomes. And then the 22nd pair, we call it gonosomes. So basically, that's it. Revision of the process of mitosis. It's advisable before you start meiosis to do a revision concerning about the process of mitosis. So guys, our next video, as we say, that we're gonna be having prizes, airtime, meals. Yes, we send you a voucher and then you buy whatever you want to buy. It can be a school bag, t-shirts. We have a lot of gifts to give out on our YouTube channel. So please don't forget to subscribe. Don't forget to subscribe, like, share, so that others also benefit from this content. And then we're gonna be having all the subjects. So we are trying to increase this. So mitosis occurs in every living organism. In the somatic body cell to produce, those are the body cells, other cells other than sperm and ovum, to produce genetically identical cells. So you can have a hole on the mouth, on the cheek, and then you need meat. So what they can do, they can cut the bum and then they put on the mouth. So you move with the bum on your face. Basically, why do they do that? Because these cells, they undergo what you call mitosis. They are genetically identical. The meat from your bums is exactly the same as the meat from your face in terms of the genetic composition. So two identical cells are reproduced, two identical cells are produced with identical chromosome number equal to the original cell. It means that the parent and the babies or the daughter cells, they're exactly the same. There is no difference. That's why when you get a wound, another meat develops. Those cells which develop, they're exactly the same. Mitosis has only one cell cycle, i.e., it doesn't have that mitosis 1 and mitosis 2. No, it only has one cell cycle. Only one cell cycle. It means that meiosis, when on the side of meiosis, it has two cell cycles, meaning that it has meiosis 1 and also has meiosis 2. So guys, let's look at the process of meiosis. Yeah, the process of meiosis was a very good introduction of meiosis. Now let's look at meiosis in detail. And this will be our next topic tomorrow. Don't miss it. Don't forget to subscribe, like, and then share the video. We will be having gifts, prizes in our next videos, starting from meiosis. Thank you very much. And also don't forget to like, subscribe, and share our new YouTube channel in the link below called The Bright Doctor. M.Saidi was giving you the lesson. Thank you.
Please use the link below to get the notes, questions, and other videos. Don't forget to subscribe, like, and share to others for our daily new videos.
Yeah, welcome back, guys. Welcome back. Today we are going to be looking at what we call the process of meiosis. So we saw the introduction of meiosis, and today we will be looking at the detailed process of meiosis, and where most questions come from in examination. Yeah, don't forget that we say that in this video we will be having some gifts for those people who are watching the process of meiosis. Don't forget that we say that meiosis is the process whereby one cell divides to form 4 daughter cells. These cells are genetically different from each other and from the parent. The process of meiosis, we say that a type of cell division that results in 4 daughter cells, each cell with half the chromosome number to the parent. So meiosis is a continuous process. It's a continuous process, but it's divided into different phases and events. So the events, for example, the first event is what we call the interphase. Interphase is not part of meiosis. You must know that interphase is not part of meiosis. It's just a growth phase, because meiosis is regarded as a reproductive process in the reproductive cells, which are determining the cells which we call the gametes. So it includes DNA replication. That's where DNA replication takes place. Remember, when you're talking about DNA, we asked a question, where does it occur? We said in the nucleus. When does it occur? It occurs before the cell divides, or it occurs during interphase. So when you say before the cell divides, it means that interphase is not regarded as a phase of meiosis. It's just a growth phase. It's like saying that you give birth to a baby, and then the baby immediately cannot give birth to another baby. Needs some time for this baby to grow, so that now once the baby grows, then is ready to reproduce. When the cell divides and then goes to the last phase, it does not divide immediately. No, it requires some time for it to grow. So during that process of growth is what we call interphase. And the things which occur in that phase, some of them is DNA replication, doubling of the DNA. Chromosomes which are single-stranded, they become double. we saw it that we have what called the unreplicated chromosomes. When they double, they become the replicated chromosomes. So when the chromosome has one thread, then we call it unreplicated chromosome, or what you call single-stranded chromosome. But when it becomes double, as we said, as you see it here, it becomes what it is called the replicated chromosomes or double-stranded chromosome. So each chromosome will consist of 2 chromatids joined together by a centromere. So a chromosome, it must have a chromatid. It must have a centromere joining it. If it is replicated chromosome, it will have 2 chromatids, and these chromatids must be joined together by a centromere. Centromere is the middle part of the chromosome. Then DNA replication. What happens in DNA replication? DNA replication helps to double the genetic material, to double the chromosome number. As I said, that we don't say to increase the chromosome number. As we ask a question, it increases the chromosome number from what to what. So we need to say that it doubles the chromosome number or genetic material. So the material, so that genetic material, so that it can be shared equally by the new cells arising from the cell division. It means that now these cells, the new cells which are going to be formed, they are going to have the content. They are going to have the content. They're going to have the chromosomes. They are going to have other cell organelles equally, shared equally, because each cell must have this content. So if it doesn't have, then it means that it will be empty, and then it won't be able to survive. So it is the function of interphase, the growth phase, to bring about the doubling of these organelles and the doubling of the genetic material, so that each cell, each new cell has a equal amount of the genetic material and also the cell organelles. If we have said that interphase is not the part of meiosis, then what are the phases of meiosis? Number 1, we have what called the prophase. Number 2, we have what called the metaphase. Number 3, we have what called the anaphase. Number 4, we have what the telophase, and then we go to Prophase II, Metaphase II, Anaphase II, Telophase II. So we are talking about prophase, metaphase, anaphase. What is it? What does it mean? Prophase means preparation. Pro means preparation. P versus P. Then you have metaphase versus middle. M versus M. And then anaphase versus apart. A versus A. And then T means telophase, T versus T. So even in Prophase II, Metaphase II, Anaphase II, and then Telophase II. So we form if we take the first letter, we form what we call IPMAT. This is in different color. It means that this interphase is not part of meiosis. That's why we don't have interphase 1 and interphase 2. We only have interphase. So it means that it's not a part of meiosis. So the process of meiosis begin with prophase, metaphase, anaphase, and telophase, whereby we say that this is preparation, middle, apart, and then they have reached the what? The terminal. What are those things which we are talking in middle, apart, terminal? What are they? We are talking about the chromosome, as you see them here. So this is trying to show you exactly what is happening. So it means that we start with one cell, the one cell divide from 2 cells, and then these 2 cells divide from 4 cells. But actually, what is really happening for this cell to divide is what you're going to look at in the details of each phase. Let's look at meiosis 1. Meiosis is divided into 2 phases, into 2 divisions, not phases. The first meiotic division and the second meiotic division. So in the first meiotic division, it will have prophase 1, metaphase 1, anaphase 1, telophase 1. And then the second meiotic division will have prophase 2, metaphase 2, anaphase 2, and then telophase 2. So the first meiosis, this is called reduction division, whereby when you talk about reduction division, you start with 46 chromosomes. At the end of meiosis 1, you end up with the 23 chromosomes in each cell. Hence, we call it reduction division. This division results in the reduction of the number of chromosomes from 46 to 23 in each cell. So the resultant gamete are haploid. It means that they have one set of chromosomes. From chromosome number 1 to chromosome number 23, they are not paired. It means that they are single haploid, half. It means that the cell has 23 chromosomes, not 23 pairs of chromosomes. All right. Let's look at the phases of meiosis. The first phase is prophase, which means preparation. The cell is preparing for whatever is going to be doing. So the first thing is chromatin network. We say that chromatin network means that entangled DNA. DNA is in form. We don't have a direction when we look at it. We don't know where it starts or we don't know where it ends. So it is chromatin network. When it condenses, it becomes thick. When it becomes thick, it forms what called a chromosome. So the chromatin network condense. When they condense, it means that now that it has formed a chromosome. Now these chromosomes, the chromosome which look alike, homo, which means the same, logos, which means structure. So it means that chromosomes which look alike, they are going to come together. That's why you're saying that homologous chromosomes are formed. It means that the chromosomes which look alike, they come together. When these homologous chromosomes, the way you see it here, when they come together, they cross over. What do they use to cross? They cross using the chromatids. The one chromatid of one homologous chromosome crosses with another chromatid of another homologous chromosome, and then the process continues. So the spindle thread starts to form. It means that now the spindle fiber, they start to form, and then once they start to form, the nuclear membrane starts to disappear. Let's look at the diagram here. So you see that here, the chromosome, they condense, the homologous chromosome. You see this one is short, this one is long. So this short cannot go with the long one. No, the short goes as with the short, the long one goes with the long one. That's the principle. These chromosomes, they come together, and then the chromatids, they cross each other. When they cross each other, we call it crossing over. That's why I'm saying that crossing over takes place. And then the point where they cross over, we call it chiasma. We're going to look at this in detail. And then now these spindle, they start, the centrioles, they start to move to opposite poles. As they move, the spindle starts to form. So once you see something like this, they have crossed like this, then you just know that yes, this is prophase. Why? Because the chromosomes are crossing over. Homologous chromosomes are crossing over. We don't have crossing over anywhere else except in prophase I. Not in prophase II. We only have it in prophase I. Why in prophase I? Because the chromosomes are still in homologous pairs. Let's look at the process of crossing over. So we have seen that the homologous chromosome, the chromosomes which look alike, they're going to line together. When they line together, one chromatid of each chromosome overlap. So it means that they cross each other. When they overlap, these chromatids, they must overlap based on the homologous pattern. For example, it means that the one chromatid from one homologous chromosome and another chromatid from another homologous chromosome, they cross over. And then when they overlap and they cross over, it means that they cross over at a point called chiasma. And then crossover takes place at a point called chiasma. If it is one, we call it chiasma. If there are many, we call them chiasmata. So when we say chiasma or chiasma, depending on the kind of English you are using, then chiasma or chiasma is a point where these two homologous chromatids cross over during prophase I. And then now the chromosomes separate. So it means that now they have formed different chromosomes. I always ask this question to different people. Are you sure your father is your father? Why am I asking you that? You don't resemble him. Actually, you don't even resemble your mother. You go somewhere, and then you find out that you resemble someone. Why? And they ask you that, hey, when you resemble someone, I know that. Why? Basically, he might be your father. But because of crossing over, this made to the exchange of the genetic material. If you came from the same father, same mother, you breastfed on the same breast of your mother, why don't you look alike? It means that this crossing over, this process of meiosis helps in bringing about variation among organisms. So it is a very important process to make humans, individuals, different from each other. And then this helps in the survival. If all of us, we are the same, then it means that we won't be able to survive. Imagine everyone is rich. Do you think that there will be someone who is working for another one? Everyone has money. Do you think I would have taught you this? No, because you don't need this. No one would have been watching. You don't need this. You have your money. What else do you want? But because there is difference among these organisms, among us, then it brings about what called competition. Hence, different people will work differently and in different environmental conditions. So that's the purpose of meiosis, to bring about variation among organisms of the same species. All right, let's continue with the process of crossing over. So these differences is being brought about by crossing over. Imagine a father producing over 100 million sperm for a single ejaculation. And then this sperm, each sperm is different from each other. What made them to be different from each other is the process of crossing over, because it forms different combination of chromosomes. All right, let's look at. So we're saying that you have one homologous chromosome and another homologous chromosome. They come together. Yes, when they come together, they overlap. The homologous chromatid overlap at a point called chiasma. Yes, if you look at ABC and then decide ABC. Yes, when they cross, now this one takes this red and then the red takes the blue. That's why you see it here. The blue takes the red and then the red will take the what? The blue. This chromosomes during crossing over, scientifically we call it a bivalent or bivalent, based on how do you write that, how do you pronounce that, bivalent or bivalent. And then after that they separate so that each chromosome has genetically different, each chromosome has different material from one another. Look, if you see that ABC, it started with ABC, but this is a small error. The small error was found on this chromosome. And then this one, ABC, but this one is a capital error. The capital error came from this blue. If you look at it here, it came from the blue. It means that the blue, the red took the blue, and then the blue took the what? The red. So because of this, it's going to cause different gametes being formed. And then, if you look at it, sometimes we can bring it in the exam like this. Homologous chromosomes, when they cross from top. So when it crosses from top, it means that you have to draw it on top. Don't draw it down. That's one of ways of losing marks if you don't show the variation correctly. So they have the chromatids cross over and then they exchange the material or the information. And then now the chromosomes now have new combination of the genes. Guys, don't forget that I told you that there is a gift in this video. Just click that link below. It will take you to a specific page on our website. And on that page, there is a code for you to obtain that airtime. Enjoy it. When you get it, when you load it on your phone, let us know that you got it. Thank you very much. Let's continue. And then you're saying that the second phase is metaphase I. Metaphase I, meta means middle. It means that now the chromosomes are always in homologous pair. Homologous chromosomes randomly align along the equator of the cell in 2 rows. It means that they align along the equator in 2 rows. One from chromosome number 1 to chromosome number 23. Another one is from chromosome number 1 to chromosome number 23. That's what it means. Each chromosome is attached to the spindle thread from each pole. This is what I'm trying to say. So they align in 2 rows. You see, they align in 2 rows, and then these chromosomes are being held by the spindle. See, each chromosome is attached to the spindle thread from each other. They do this arrangement randomly. So the key point is homologous because they're in pair and then each other. You see, this one cannot come here. No. They must be together like this, homologous chromosomes. Yes, that's the key point. And then they come here randomly. Remember, we have 23 pairs, but we're just showing you 2 pairs. Yes, they are here randomly. So it means that another one can be here, this one can be on top. So this set can be on top, this one can be down. Or this chromosome can be this side, this one can be this side. That's what we call randomly. So when they separate, we're going to form different cells. So each chromosome is attached to the spindle fiber or spindle thread from each pore. That's what we are trying to show you, that these threads are attaching the what? The chromosomes from each pore. So after that, once they are done with this, then they start to move apart, which you call anaphase. So in anaphase, the spindle thread shorten, they try to pull. When they shorten, they pull. It's like when you are a bodybuilder, when you carry something, the muscles shorten. When they shorten, they pull. Yes, so they pull the homologous chromosome to opposite poles. And then the chromosomes, what happens to this chromosome, the homologous chromosome pair, then they do what? They separate. The chromosomes of the homologous pair, they separate. It means that now they move to opposite poles. This is what is happening. So now they move to opposite poles. And then now, when they move to opposite poles, now they reach the pole. So in telophase, each chromosome reach at the pore of the cell. And then once they reach the pore of the cell, the cytoplasm and the cell membrane divide. When the cytoplasm and the cell membrane divide, that process is called cytokinesis. We call it cytokinesis, division of the cytoplasm. And then the 2 haploid cells are formed. 2 haploid cells are what? Are formed, and then the nucleus or the nucleolus that dense body in the nucleus, they reform. Then it reforms, and then lastly, the new nuclear membrane forms, and then now the cell appears to be a full cell. So this is what I'm trying to say. Yeah, so now we form 2 cells. Now these cells, cytokinesis took place, and now the nuclear membrane start to reappear, and then the cell goes back as if it is in prophase. So now when it reaches there, now each cell, each of it will undergo meiosis II. It means that now this one is going to go under meiosis II, even this one is going to undergo meiosis II, so that you form 2 here and 2 here. So when you add them, you form 4 cells. So when you go to meiosis II, what happens? Now, prophase II, you go to prophase II. When it's meiosis II, prophase, it becomes prophase II. The 2 must be there. If you don't write it there, we won't give you a mark because we don't know which prophase is it. So prophase II, what happens? The nucleolus, remember we said that it appeared, it reappeared. So now the nucleolus and the nuclear membrane, they disappear again. But now here we don't have crossing over. So the new spindle fibers are formed. Yes, they start to form, and then the chromosome become visible. It means that they condense, and then it becomes visible. And then now, once they become visible, now it's ready to undergo metaphase. They go to the middle of the chromosome, the cell. So now you see it here. Now it is forming like this. And then now they are ready to move this thing to attach themselves to the chromosome. But now we don't have homologous chromosomes. We have chromosomes which are not homologous. It means that crossing over cannot take place because it already took place before. Yes. So now, in metaphase II, the individual chromosome line up here. Individual or one chromosome align along the equator singly or individually. So the other side, we say that chromosomes, they align along the equator in homologous pair and randomly. Here, they align along the equator singly or individually. So you have to know the difference. So sometimes you ask you the difference in meiosis I and meiosis II. You can use the process to differentiate between the 2. So at the equator of the cell, with the centromere attaching the spindle fiber, or centromere attached to the spindle fiber. And then now, this is the picture. If you rotate it, you will see that these chromosomes are in the middle. You see, they're in the middle. If this is the pole and this is another pole, they are in the middle. So this one becomes the equator. Remember, I said that equator is an imaginary line. It's an imaginary line. It's a line which we just imagine, but the line is not there. It's a imaginary line which divide the cell into 2 equal parts. It's the same thing, equator of the earth. Imagine a line running from east to west and dividing the earth into two equal parts. We're just imagining, but the line in actual sense is not there. We just imagine that that's the center of the earth. So we just imagine that's the center of the cell. So in anaphase, once they're in the middle, they have to separate to opposite poles. So in anaphase 2, you have spindle fibers start to form. So the spindle fibers, they contract. When they contract, it means that now they pull the chromosomes apart. But now, because they are single chromosomes, they are not homologous. Now it's this the chromatids which are going to separate. Remember, chromosome is divided into two chromatids. Now, the one chromatid is going to go one side, and another chromatid is going to go to another side of the cell. So this is what I'm saying, that now the chromatids move to opposite. It was together here. Now this one pulls it this side, and this one pulls this side. Check the direction of the. It's not facing this way. It's facing that way, because the force of pulling is coming from this side. So that's why it's supposed to be like that. When you're drawing, make sure that you take caution when you're drawing it, so that you draw it properly. And check about the shadings. Also, the shadings are very important when you are doing it. The daughter chromosomes reach the poles once in telophase 2. In telophase 2, the daughter chromosomes reach the poles. When they reach the poles, we don't call them chromatids anymore. We call them chromosomes. Actually, that's what we call single-stranded chromosomes or unreplicated chromosomes. So it's supposed to get time, so that this time allows these chromosomes to grow, so that now it becomes double-stranded again. That's why we need what you call interphase. So new nucleus then forms. It means that the nucleolus also come back. It means that it reforms. Then the cell membrane of each cell constrict, and then the process of cytokinesis occurs. When the cytoplasm divides, now the process of cytokinesis occurs, and then for haploid, it must be haploid. Daughter cells are formed, and then now each daughter cell have the chromosome number to that of the parent. So that's why you're saying that meiosis is the process whereby one cell divide to form 4 daughter cells which are haploid, or which have half the chromosome number to that of the parent. And these chromosomes are genetically different because of the crossing over which took place. So the daughter cells are genetically different, as I've just said. And then these are the cells which will form. We form 4 daughter cells, and then you see, we don't call these ones chromatids. We call them single-stranded chromosomes. Watch out this before you write your SBA. So the importance of meiosis. What are some of the importance of meiosis? Let's look at the importance of meiosis. Shall start from there with the importance of meiosis. M.Saidi was giving me the lesson. Thank you very much.
Yes, good morning, good evening, and good afternoon, my dear students. If your notes are not up to date, like the notes you are using, or you don't have the notes yet, there is a link below in the description. There is a link below. For that link, you will be able to access these notes so that you can prepare yourself. Let's look at blood grouping. Blood grouping. Do you know your blood type? All right. Different blood groups are as a result of multiple alleles. When you talk about an allele, we say that it is an alternative form of a gene. For example, let me talk about height. For example, you might be tall or you might be short. You have the allele for tallness or you have alleles for shortness. So it means that if it is a character is height, but this height has 2 alternative forms. The gene is height, but has 2 alternative forms of it, whether you are tall or you are short. So that's what we say, that allele. So it is an alternative form of a gene. But now when you say multiple alleles, it means that it's more than two. So blood group, it is controlled by multiple alleles. It has more than two alleles. Three or more alternative form of a gene, that is the allele. Yes, you have a gene, but it can exist in different forms. Yes, that can occupy the same locus. That's what we call the multiple allele. Three or more alternative form of a gene that can occupy the same locus. When you say locus, locus is the location of the gene on the chromosome. So it is the same location of the gene on the chromosome, but you have different or many alleles in that location. So you're saying that, however, only two of the alleles can be present in a single organism. Why should it be two? It's because you have two parents. You have the father and you have the mother. So one allele must come from the father and one allele must come from the mother. The alleles the father has and the alleles the mother has, so the one of it will come from each parent, and then they combine so that they form your alleles. So that's the meaning that, however, two of the alleles must be present in a single organism. For example, if you look at a blood group, you have the what called the phenotype and you have the what called the genotype. The phenotype, sometimes it is the blood type or the blood group. The blood group, that is the phenotype, what you see. Because when I ask you which type of blood group are you or do you have, or you say I have blood group A, so that is what we can see, at least, because phenotype means what is outside. And then a genotype, but what is the composition of that blood group A? Is it homozygous or it's heterozygous? So that is what called the genotypes. You are describing your blood group in terms of genes, the kind of genes you do have. So we are saying that you have blood group A, you have blood group B, you have blood group AB, and blood group O. Blood group A can be in two different ways. You can be homozygous or you can be heterozygous. Homozygous, homo means the same. So when you say homozygous, it means that you have the same kinds of alleles. You have this A and A, you see. And then heterozygous, you have this A, which is the dominant, and then a recessive one. Blood group B also has two kinds of genotypes. You have homozygous blood group B, it is the same kind, or it has heterozygous. Hetero means different. Then you have blood group AB. This one, it only has one option, which is AB. And this is co-dominant, whereby the two alleles are expressed equally in the phenotype. And then you see you have blood group O. Blood group O also has one option, that is two recessive alleles. So it means that if you have blood group O, it means that you have a recessive allele for blood type. Now you need to know this. You need to understand this before you go for your exam. When they say blood group A, these are how many, they can ask you a question, how many phenotypes do blood groups have? You have how many phenotypes? 1, 2, 3, 4. There are 4. How many phenotypes? There are 4. How many alleles? How many alleles controlling blood group? You see, it's being controlled by 3. Blood group, allele A, allele B, and allele the small I. So those are the 3. How many genotypes do we have in blood group? You have 6. 1, 2, 3, 4, 5, 6. There are 6. So the genotypes, there are 6. The phenotypes, there are 4. And then alleles, there are 3. We're going to look at that one in detail. So we are saying that blood groups, you have A, you have B, you have AB, and you have O. These blood groups are being determined by the red blood cells. So which kind of antigens do you have? So you see that blood group B Blood group A has this kind of antigen, blood group B has this kind of antigen, blood group AB you have this kind of antigen, blood group O you have this kind of antigen. So this one makes which blood group who donates to who and who receives from who. So because of this kind of antigen, if it is not compatible, then it means that you can't receive or you can't do it. That's why blood group O, it can donate to anyone. Why? Because it doesn't have the recognition sites. And then blood group AB, it can receive from anyone. Why? Because it has both for this one and then this from this. So can receive from anyone. Why? Because it has the universal receptors. All right. So we are saying that alleles A, B, and I in different combination results in 4 different blood groups. That's what I've explained here, that you have 4 different blood groups. You have A, B, AB, and O. So you have 3 alleles. The first one is A, the second one is B, and the third one is I, small I. These alleles must be written like that. Don't modify the writing. Don't shade it. Don't put an O. Some people, when they are writing, they write I and then they put a small O on the top. That's not I. It must be I and a dot on top. You have to be able to write. That's how you are supposed to write it. However, some books, they can show that, yes. So with this, our standards, we don't want you supposed to write the I the way it's supposed to be. So here you can see that you have 3 different alleles, that is A, B, and small I. Then you have 4 different types of blood groups, A, B, and O. So it means that all these ones are being controlled by these 3 alleles. These ones must combine to form these 4 blood groups. Then here is the table trying to describe what I've been trying to explain. So this is the blood group, sometimes call it the phenotype. Yes, what we see. These are the alleles, sometimes we call them genotypes. Why? Because the alleles are genes. So genotypes is description of an organism based on its genetic composition. So you have A, you can be homozygous A or heterozygous A. You can be homozygous B or heterozygous B. You can be codominant AB, or you can be homozygous recessive. So this is homozygous dominant. This is also homozygous dominant because this is the dominant allele, and then O is a recessive, so it's a homozygous recessive. So make sure that you understand these concepts because they will ask you. So A is, if your blood group A outside, yes, we know that your blood group A, but genotypically, which kind of blood group are you having? Is it homozygous or it is heterozygous? So you need to understand those concepts. That's why you find out that a person of blood group A marries a person of blood group B, and then they produce a kid of blood group O. How is that possible? It is possible, and some people used to deny their kids, yet they are their kids. That me, I'm blood group A. Yes, you're blood group A. The mother is blood group B. Yes, you're blood group B. How comes that I have a kid of blood group O? It is possible. If I have a mother of blood group A, heterozygous, and a father of blood group B, heterozygous, I can produce blood group O. Why? Because this small I, or the recessive I, and a recessive I from here, when they combine, they form 2 recessive I's. Remember I said that you obtain these alleles from your parents, one from the father, one from the mother, and then you form your genotype. So it means that 1 I from the mother, 1 I from the father can have 2 alleles combining to form blood group O. So it is what? It is possible. So let's look at some of the problems. When you talk about problems, we look at some questions of blood group. Susan, with blood group A, with blood group AB, marries John with blood group O. What will be the possible genotypes of their offspring? What could be the possible genotypes of their offspring? Then let's look at it. So in this case, you're supposed to find out what is the genotype, what's the phenotype, and what's the genotype. So now you write P1, F1, yes, and then P1 means first parent, parent one. So meaning that the first generation. So it means that you're saying that always the phenotype is obtained from the question, always from the question. Look at the question, read it nicely, you'll find the phenotype there. So you're saying that Susan of blood group AB, Susan of blood group AB, yes, marries John of blood group O, marries cross means marries John of blood group O. This AB is heterozygous, has one option. O also has one option. So that one is, this question is just open for you, just to get marks for Mahala. So we are saying that genotype, the genotype, because we know that blood group A, blood group O looks like this, blood group AB looks like this. So these are genotypes. So you need to know the genotypes. So now we are saying that the genotype of blood group AB is like this, genotype of blood group O is like this. So these two, meiosis must take place. When meiosis takes place, so it produces what called a gametes. So meiosis must take place. You must write this one there. It divides these genotypes into two gametes. So we are saying that the gametes are being formed. The first one is your blood group A, yes. Blood group B, yes. Blood group B is A from here, and then B comes here. Then O, they divide one from here, and then one from there. So they form 4 of them. So after that, since fertilization must take place, we need this. Fertilization takes place. When it takes place, you have now the allele. You have these two, you put them on top, and then these two, you put them down or here. You can reverse them. Put these ones here, put these ones here. It still is the same thing. You don't need to cram. As long as one, put them here, one, put them there. Yes. So now this crosses with this, because when I draw a line here and a line here, yes, they meet here. You see? So now this and this, you have A, capital A, small I. Then you have this and this, you have I, capital B, and then small I. Then this, when I come here in this box, this with this, so it's gonna be capital A and then small I. So it must be, you begin with a capital letter and then you end with a small letter. So you have this and this, so you have capital I, small capital B, and then small I. So that is the genotypes of the offsprings. So the first F1 generation of F1 genotypes, you're gonna have A, this one, and this one. These are the genotypes of F1. Why not others? Because this and this is the same, so 2 of them, and then this and this is the same, 2 of them. So basically you have, the ratio is 1 to 1. 2 to 2, when you reduce it, it goes to 1 to 1. Then the ratio is 1 to 1. The genotype, the phenotype, though they didn't ask you, but you can write it. You have blood group A. Yes, this is blood group A. Don't say blood group A homozygous or heterozygous. No, we don't see the homozygosity or heterozygosity in a person. You can't see that unless you go to the genes. So what we see outside, it is blood group A, and what we see outside is blood group B. You don't need to describe what is real inside. Just say blood group A, blood group B. And then basically that's it. Now let's look at how do we mark. When we are marking, we come here. Sometimes there is a compulsory mark, so it depends on the examiner. So we are saying that P1, F1, we give you a tick. We give you a tick there. They're saying that meiosis, fertilization, the word meiosis and fertilization, the two, just writing them, you get a mark, even if you don't have anything here. As long as you write them like this, you get a what? A tick. Then It means that don't forget to write them. Then you have genotype, we can give you a tick. Yes, phenotype, you can give you a tick. See? And then writing the gametes, you can give you a tick. And then here I also give you a tick. Yes, and then here you also give you a tick. So if it is out of 5, and then for example there is a compulsory mark, for example, or they say that according to the question, the genotypes, so you must write the genotypes. Yes, the genotypes, if that is the compulsory mark, even if you get 7 here and you didn't write this, and the question is out of 5, you get 4. Why? Because you didn't get the correct answer of the compulsory mark. We first count the compulsory mark, then after that, then we can count any other. So make sure that when you are writing, at least maximize, do whatever you all the steps, write all the steps, so that if the compulsory mark is there, you can get it. So this is the practice question. Let's look at the practice question here. It must be guided by the teacher. So now I'm your teacher. All right, Cabello of blood group A marries Carabo of blood group B, and they produced children of blood group O. Use the genetic cross to show the phenotypic ratio of their offspring. They want the phenotypes, so the phenotypic ratio. In this case, what we are supposed to do is go back to the table and see they said blood group A marries blood group B, produce kid of blood group O. I think I explained this. So it means that it's gonna be heterozygous blood group A and the heterozygous of blood group B, because when I try to cross them, they produce blood group O. Then you come back to the question. So it means that these parents, they were heterozygous to produce blood group O, which is a recessive. So now you have to do a genetic cross, P1F1 phenotype, then you tell us genotype, you tell us meiosis must take place to produce the gametes, fertilization must take place, because now you have the gametes, you have the sperms and you have the ova. So now the fertilization must take place. When they takes place, then you produce the offsprings. The offsprings will be genotypes. Those genotypes, then convert them into phenotypes, and then find how many blood group A, how many blood group B, how many blood group AB, and how many blood group O. Then they're saying that how many alleles controlling blood groups. How many alleles? We saw this one. How many alleles? There are 3 alleles. Blood group. Allele A, allele B, and then allele I, small I. Yes, we saw them. And then how many blood group types do humans have? They have 4. A, B, AB, and O. Basically, that's it. Next class, we are going to be looking at dihybrid cross. Don't forget to subscribe if it is your first time to attend this class. See you next time. Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. We are saying that genetics, when we talk about genetics, we are talking about what we call genes. Small sections of DNA, and these sections, they have a certain character they are controlling, is what we call genes. So the study of inheritance, the study of these genes is called genetics. Basically, in this topic, these are the things you need to cover. I'm going to cover the following subtopics in this topic. The first one is concepts of inheritance, the terminologies. You need to know the terminologies. Monohybrid crosses, crosses which involve one character, mono, one character. Sex determination, what is the percentage chance for you producing a girl or a boy? How do you determine the sex of a kid? Sex-linked inheritance, the characters which are linked to the sex. Blood grouping, which type of blood group, and then inheritance of blood group. Dihybrid crosses, when we talk about dihybrid crosses, we mean inheritance of two characters. Di means two. Genetic lineages or pedigree. These ones are tracing characters in your family. In most cases, in this case, you're going to be looking at the disorders which can be inherited by the family. So you have mutation. When you talk about mutation, it means change in a part of DNA. It could be the whole chromosome can change, or it can be just a small section of DNA changing. So we're going to look at the mutations. Genetic engineering. Genetic engineering, it means that you're trying to manipulate. So you try to manipulate with the genetics or with the genes of an organism. Then you have a paternity test. Here you're trying to do the testing of the parents, father, mother. If there is any subtopic you don't understand, then it means that you are still lacking, you are susceptible to lose marks because you don't know which part of genetics they are going to bring. You can bring any part from genetics. So you don't know exactly which part they're going to bring. And if you don't study that part, then it means that these marks which are allocated on genetics, you have lost some marks even before you go to the paper. However, by chance, you can find out that the part you don't know, it has not come, and then you can answer those which you know. But I don't encourage that. So let's look at the introduction. Father of genetics. When we talk about father of genetics, we are talking about Gold Mendel. He did a lot of work on inheritance, and he discovered a law which we call law of inheritance. He concluded a gene come in pairs, like when they are, for example, height is being controlled by two parts of genes. When we talk about genes come in pair, a character like, for example, height, size, skin color, they're being controlled by two kinds of genes. So they come in pairs and are inherited as distinct units. It means that one character is not interfering another character during inheritance. Each character is inherited differently, from one from the father and one from the mother. It means that each parent is supposed to give one part of the character. Remember, there are supposed to be two, so it means that the first part comes from the father, the second part comes from the mother. That's what he is saying. So let's look at some of the concepts of inheritance, meaning that the terminologies. So chromatin. When you talk about chromatin, it's a substance within a chromosome containing DNA and a protein. Basically, when you talk about a chromatin, chromatin is DNA. But in this case, this DNA is like threads. Yes, it is in coiled form. When it coils, then it forms what you call a chromatin. Then number two, you have what you call chromosome. Now, when the chromatin condenses, when it shortens, when it becomes thick, it forms what you call chromosome. So chromatin and chromosomes, they are the same thing, but in different versions. So you can say water in ice form and in liquid form. So it's the same thing, but it's in different phases. So we are saying that thread-like structures of nucleic acids and are found in the nucleus carrying genetic information. So chromosomes are the thread-like, they are like threads crossing each other. And these chromosomes, they carry what called the genetic material. You look like your father because of the genes your father gave you. Then, if you look at this structure, look at this, you might think that this is noodles. Yeah, you think that these are noodles or spaghetti. Yes, but if you condense it, yes, so if you shorten it, you squeeze it, you condense, it forms what you call chromosomes. So when they cross, they look like this. So when you talk about chromosome and the chromatin network, they are the same but in different phases. You see, you can see that what is here is almost what is here. Small ingredients just to make them to be in this shape are being added just to make them to be chromosomes. But what they carry inside, the most important thing, what do they carry is the same information as chromatin network. But they form chromosomes during cell division. What about genes? Chromatin network, chromosomes, genes. Genes are just small sections on the chromosome. So if you just break down the chromosome or the DNA, you form different portions. Those different portions, they form what called genes. And then you have, we are saying that this is a small portion of DNA coding or controlling a particular character, a particular protein. So it must be these characteristics you have. You are dark-skinned, you are light-skinned. What causes that? It is genes. And then when we talk about allele, alleles are genes. Alleles form chromosomes. Alleles form chromatin network. But chromatin network, if you look at chromatin network, you look at chromosomes, you look at genes, you look at alleles, they are the same things. All of them is the same thing, but at different scale. I'm gonna give you an example. When you talk about that, where do you stay? You say that I stay in South Africa. Where exactly? Then you tell me that I stay in Northwest. Where exactly in Northwest? You tell me that I stay in Rustenburg. Where exactly in Rustenburg? So you keep on reducing the scale. Chromosome, genes, alleles, they are the same but at different scales. So when you talk about allele, it's an alternative form of a gene. You can be tall or short. So an alternative form is called an allele. So we are saying that alternative form of a gene found at the same locus. Locus means the location of a gene, the place of a gene, the position of a gene is what we call a locus. We're gonna bring these things in terminologies. And then please don't forget to give a correct answer. So here is a chromosome. A chromosome is here, but you have different parts, different locations of these chromosomes. So these parts is what you call the alleles. So these alleles may be for height, for size, for color, for blood group. They are supposed to be found on the same position of the chromosome. This side and this side is the same thing. So we call them alleles. So are alternative forms of gene. Then we talk about a dominant allele. So because you have two forms of alleles, one can be dominant, another can be recessive. And both of them can be dominant, or both of them can be recessive. So when we talk about dominant, it means something which suppresses another thing. Yes. So we're going to say that an allele that is expressed in the phenotype when found in heterozygous state. When we talk about heterozygous, it means in different phases. One is like this, one is like this. So it means that this capital letter suppresses this. That's why I say that a dominant allele is that one which suppresses another allele. So this capital letter will suppress the small letter. Then what you call the recessive allele. This one can only express itself in absence of a dominant allele. So if this one is absent, then automatically this small t will also express itself. So we are saying that an allele that is masked in a phenotype when found in a heterozygous state. So it can only show itself in a homozygous. It is only expressed in a homozygous condition or homozygous state. Here is an example. You have homozygous dominant. It means that, you see, they are the same. Homo, I say that when you talk about homo, means the same. Zygous comes from what? Zygote. So it means that this zygote has the same kind of allele. Heterozygous, it means that this zygote has different kinds of alleles. So you have capital letter and small letter. And then homozygous, still the same type of alleles, but these alleles are recessive. So it means that it is the same kind, homo, which means the same. So same, but they are supposed to be recessive. Then when you go to phenotype, when you talk about phenotype, it means the physical appearance of an organism. You are expressing an individual in terms of what you see. In most cases, when you bring a question concerning about a cross, they're going to ask you the phenotype. The first question, the first part, you are supposed to write the phenotype. We get these answers from the question. Is it clear? So we are saying that physical appearance of an organism is determined by the genotype. For example, tall or short. You can be tall, you can see that a person is tall. You can be short, you can see that a person is short. You can be light-skinned, a person is light-skinned. You can see them. And then what about genotype? Here you are describing an organism in terms of what? In terms of genes, genotype. The gene type. Which kind of gene type do you have? So here is genetic composition of an organism. So you know you are tall, but which kind of tallness? You might be tall, but homozygous tall or heterozygous tall. You have an allele for shortness. You can be short, but you have an allele for tallness. You see two people who are short, they produce a tall kid. Two people who are dark-skinned, they produce a light-skinned kid. Two people who are light-skinned, they produce a dark-skinned kid. And then a guy can say that, yeah, this is not my kid. It's whose kid? All right. So here is another example. When we talk about this, describing this in terms of the genes is what called the genotype. Then also this is the genotype, and also this is what? Genotype. So I will say that this is homozygous dominant, this is heterozygous, and then this is homozygous recessive. So I'm describing these flowers in terms of genetic composition. So then when we talk about homozygous, I've been talking about this. Homo, which means the same zygous from the zygote. Then it means that this zygote has the same kind of genes. Two alleles, two identical alleles for a particular characteristic. Homo, same, can be homozygous dominant or homozygous recessive. When we talk about hetero, when you say hetero, it means different. So the zygote has different kinds of alleles. For example, capital letter B and then small letter b. So they are different letters or different alleles. If you look at this picture, you'll find out that homozygous, you have this. They are found on the same locus, same location. This one, and then this one, and then this one and this, you see. While if you look at heterozygous, same location but different letters. Same location but different letters. So this is homozygous recessive, this is homozygous dominant, and then this is heterozygous. For the case of heterozygous, we don't have recessive and dominant, just say heterozygous. A monohybrid cross is a cross which involves one character. You are right, it involves one character. But now you said pink and white, it still is fine. The answer was correct. But where is the character there? The character is color. That's the character. But this character has two alternative forms. The blue you talked about and the white you talked about. It's either blue or it's white. So those are the what called the alleles. So the character is the color of that individual you talked about. And then alleles are the different types of colors you have talked about. You understand?
Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel.
We have three types of dominancy: complete dominancy, codominancy, and incomplete dominancy. So let's look at them in detail. When we talk about complete dominancy, when you are dominant, it means that you have powers over another individual. So it's a genetic cross where dominant allele masks, blocks the expression of the recessive allele in a heterozygous condition. So in complete dominancy, you have heterozygous condition. So you have what you call the incomplete. When we talk about incomplete, it means that yes, the dominance is there, but it is incomplete. So it means that one brings 50 and then another one brings also 50. So you're saying that it's a genetic cross where none of the allele is dominant over the other. Neither of the allele is dominant over the other. It means that they have equal powers. The president of South Africa and the president of America, both of them, they have the same powers in their countries. So it means that neither of the president is dominant over the other. When it is the meeting, the president of South Africa will have the same contribution like the president of America. That's the meaning. Not that, no, because America is a superpower, now South Africa, no. Both of them, they are presidents. Neither of the two is, they are dominant, yes, they are dominant, but neither of them is completely dominant over the other, over each other. Although your country is big or small, still you have the same power as presidents. For example, you have white and you have white and red. When you cross them, they form what called the intermediate. Intermediate means that neither of the two is dominant over the other. When you cross them, they produce the intermediate. It's not white, it's not red, but it is in between there. It produces a new character. So the character which is being produced is new. That's the meaning. Yes. But here you have the one character from the parent. Black and white, you either produce white or you produce black. Here when you cross, you don't produce white, you don't produce black, but you can produce gray, which is in between the two. It's not white, it's not black, but is in between. That's the meaning, intermediate. And then you have the co-dominant. Co means together. Co means together, means two things are existing at the same time. They have the same power, they have expressed themselves at the same rate. For example, you have white and black. When you cross them, you produce a mixture of white and black. They are co, they are together, they are expressed outside or in the phenotype together. You have the white and the black, they are living together. That's the meaning of co-dominance. It means that both of them are dominant and their dominance is being expressed in the phenotype at the same time. So here you're saying that a genetic cross in which both alleles are expressed equally in the phenotype. Let's look at these examples. You have complete dominancy. Dominance allele completely masks the recessive one. It means that you have here, you have the red and the pink, and then you produce red. It means that you have one character being produced in the offspring, either red or pink. One must be produced. Then in incomplete, you have red and white. You don't produce red, you don't produce white. You produce a new character, which is pink. So neither of these is completely dominant over the other. So they produce an intermediate between red and, it means that this one has brought 50 and this one has brought 50, and then I form an intermediate color. Then if you look at the codominance, it means that the two must be there. If you look at this color and look at this color, the red and blue, when you cross them, you produce a mixture of red and blue. Here you produce a mixture. It means that red is completely dominant and blue is also completely dominant, and they express themselves in the phenotype of the offspring. While in incomplete, this is an intermediate, because this one has brought me 50 and also this one has brought me 50. This one, you don't produce neither of the color from the parent. You produce an intermediate. You produce a new color. And then this one, codominance, together, it means that both of the parents' color is being expressed in the phenotype. I think that's how you can differentiate among the three. Then let's look at one of the questions involving the dominance. So they are saying that An allele for tallness is dominant over allele for shortness. A man who is homozygous. So I know that homozygous, tall, marries, and then they said here tallness is dominant over shortness. So it means that this guy is homozygous dominant. So marries a lady who is homozygous short. And then short, they say that is what is recessive, or it is recessive. And then using a genetic cross to determine the possible genotypes and phenotypes of the children. So what am I going to do? P1, parent 1, phenotype. Tall guy marries a short lady. You get it from the question, as I told you. From the question, a man who is homozygous, tall. This word homozygous will help us in the genotype, to identify the genotype. The word homozygous, heterozygous, only is important in showing which kind of genotype are you having. You don't put it in the phenotype, because you can't see that this person is heterozygous or is homozygous using the phenotype. You can't do that. You only know that this person is tall, this person is short. But if you go into genetic composition, you will know that, oh, you are homozygous tall or you are homozygous short. That's why we don't add the word homozygous or heterozygous here. So we are saying that a man who is homozygous, we have seen tall, yes, marries a lady who is homozygous short. So we go to genotype. Genotype. They said homozygous tall. Letters are supposed to be the same. And then you have homozygous short. Letters are supposed to be the same. Why they said it here? Homozygous short. Then you go to meiosis must take place. When meiosis takes place, it divides into two. Even here, it divides into two. What do you form here? You form what called a gametes. So it means that this T is going to come here, this T is going to come here, this T is going to come here, this T is going to come here. And then fertilization must take place. It means that they must cross. You can use a cross or you can use a Punnett square. Either of the two is correct. And then they cross that, crosses this, and then that crosses that. So this one crosses these two. That's one is done. Then I go to this. This crosses with that, and then this crosses with that. And then I get the results, which you call the F1, the first period generation. So when I cross, I get this. So it shows that all of them were tall. But which kind of tallness? The tallness, if you are describing in terms of genes, it is heterozygous tall. So, or you can use this kind of option. Don't use two, because we want one. So then the genotype, it means that all of them are capital T, small t. Which kind of phenotype is this? Because I have the capital letter T, then it means that this individual is going to be tall. So which kind of phenotype? All of them are tall. I don't say heterozygous tall. I don't see the heterozygosity in the phenotype. I don't see that. So I just have to say they are tall. And then the individual of F1 all displays the dominant characteristic. It means that all of them are what? Are tall. In incomplete, you saw that in complete, you have the capital letter and the small letter. You see? You have capital letter and small letter. But in incomplete, neither of the allele is completely dominant over the other. Then it means that we don't have a small letter there. So an allele for red and allele for white is also dominant. When the homozygous red crosses with the homozygous white, it means that both of them are dominant. Using a genetic cross, determine the possible genotype and the phenotype of the flowers produced. I have P1 phenotype red. I get it from the question. Red crosses with the white. They're saying it. Red, white crosses with the red from the question. So I always get that mark from there. You have 3 marks free without even thinking. Then genotype. Now I have to come here and then look for these words. They're saying when a homozygous red You see, homozygous red, same letters. Homozygous white, same letters. Then meiosis takes place. When meiosis takes place, it also splits into gametes. So gametes, when they are formed, fertilization must take place. So you can use that method, or you can cross either of the two. So you bring these ones, you put them there. You bring these ones, you put them there. Then you cross them. You cross them. Or you can bring these ones here and then bring these W's on top. Either way, it's correct. And then F1 generation, if you look at them, all of them, they are capital R, small w. Capital R, small w. So the genotype, all of them is capital R, small w. And then the phenotype, which kind of phenotype is this? Capital R, small w. If you go back here, capital R and small w, it's not red, it's not white. So it shows that it is an intermediate of the what? Of the two. So it gives me pink. So if it gives me pink, it shows that this is incomplete dominance, whereby I produce a different color. And then offsprings have an intermediate form of trait of the parent. So that's how I can describe them. So note that in combo dominancy, you don't have small letters. Then codominance is the same as in combo dominance, but the only change which is there is because you produce a mixture of the two colors. So they're saying the cow with white fur, capital W, were crossed with a bull with red fur, capital R, as shown in the diagram, using genetic cross to show how the run was got. So how did we get this? So phenotype, P1 phenotype, I have to look for it. Okay, well, white fur, so I say it's white fur crossed with red fur. So it means that red is crossing with white. Red is crossing the white. Here they didn't talk about homozygous, heterozygous, but the statement, the word "ron" shows that it's a mixture. Ron means a mixture of two colors. So it shows that these two were homozygous, so that they gave rise to this ron. So now I have to know that capital R and the capital R, small w, sorry, capital W and capital W. Then when meiosis takes place, I produce gametes, which gametes they undergo fertilization. I give you a tick and this and this. So it means that you have scored a tick there. And then I cross them. You can use the cross or you can use this method. Either way is correct. And then I get the F1. So P1 F1, I give you also a tick. So please don't forget those. Don't forget this. Don't forget this. Don't forget this. 3 marks are there for mahal, for free. So we are saying that the genotype is this. Why? All of them are like this. Capital R, small, capital R, capital W, capital R, capital W, all of them. So it means that this is not red, it's not white. What is it? It's a mixture of red and white, which we call ron. That's the meaning. And this is how they wanted you to do that question. It's not difficult if you look at it. In codominancy, you produce a mixture of the two colors, while in incomplete dominance, yes, you produce an intermediate color, but the crossing is exactly the same. So we are saying that both alleles are equally dominant and are expressed in the phenotype. Okay, this is just trying to do a summary. Proportion and the ratio of the genotypes and the phenotypes. Sometimes they ask you to do the ratio, sometimes they ask you to do, yes, the ratio. So when you talk about the ratio, it describes the number of times a genotype would appear or the number of times the phenotype would appear. So for example, if you look at this is capital R, capital R, capital R, capital R, capital R, small r, capital R, small r, and then small r, small r. So I just have to say this is 1, and then this is 2, and then this is 1. So I'm going to say 1, to 2 to 1. 1 is for this, and then for 2 for this, and then to 1. That's basically what they want in the ratio. And then if phenotypic ratio, you see how many, if this is, let me see, did they give you, they didn't give you the color. For example, if this is red and then small r is white, then it means that this one is going to be red. This one is going to also be red. Why? Because it is having a dominant allele, which is masking this. So it's going to be red 1, red 2, red 3, and only 1 white. So I'm going to have 3, 2, 1. That's the meaning here. Actually, it's the same thing I've given you. Red is 3, red, and 1 white. So the ratio is going to be 3, 2, 1. Note, let students practice different examples with the crosses. So it means that when I finish here, I'm going to send you some questions for you to practice. So basically, that's it. And then if you look at sex determination, they're saying that the evidence of evolution. Number 1 is biogeography. As you hear the word, bio means life. Geography means distribution in different locations. So when you talk about, it means that you have life which has been distributed in different locations. So look at these camels. They have the same ancestor, but they have gone to different areas. When they went to different areas, this one here, this one there, this one there, this one there. So they modified. So this helps, or it brings about organisms to adapt differently. So if you look at them, you will know when and how did these species evolve. Why is it that you have different colors in humans? So you can use by geography because they are located in different environment. People from hot areas, you'll find out that they have different skin color. So that can help us to know how did these organisms evolve. Number 2 is fossil records. Fossils remains of living organisms. When you die, you become bones. So the bones will become remains. And if it happens that it takes for a long period of time, and then other generation come and then they try to find your bones, and then maybe the generation is different, then they will study them and they say, oh, okay, in this time, people who lived on this earth, they were like this. Just using your remains so that they can now predict how life was by then. So that's what's called the fossil records. If you look at this, it's sure that the layers of the earth, the lower layer will have the simplest form and the upper layer will have the more complex form. It shows evolution because increase in the complexity from simple to complex. So it means that the simpler one, they existed before, and then the more complex one, they existed just recently. So if you look at this horse, you find out that the skull you find in different strata of soil, different strata. Strata means layer in science. When you say stratum, it means layer. So when you say strata means layers. So different strata, you'll find out that simple will be in the lower and then the more complex one will be in the upper layers. So fossils remains have been found in rocks of all ages. Fossils of the simplest, where do you find them? You find them in the oldest rocks. Simplest, oldest rock. And then fossils of more complex organisms are found in newest rocks. You find them here, you see. And then you're saying that this supports the Darwin theory of evolution, which states that simple life forms gradually evolve into more complex ones. Like simple life forms, they evolve into a more complex one. That's why I said that you find out that you are more complex than your parents. Yes, you are more complex than your parents. It means that you evolve from simple to a more complex organisms. You have another evidence which shows that evolution really took place, which you call the comparative anatomy. When you talk about comparative anatomy, this sometimes you call it modification by descent. You modify as you shift from one generation to the next generation. If you look at this deals with similarities and the differences in anatomy. Anatomy is the body structure. How do you, if you look at your body, how is the shape of it? Yes, if your shape is not the same as that of a cow, it's not the same as that of a pig. Yeah, they are not the same. But if you look at the anatomy, the inside part, they might have some similarities. So if they have some similarities, then it means that these organisms, at least they had some line of ancestry. So they have some common ancestor somewhere, but they diverged. They modified as different generation came up. So when you talk about organisms with the same ancestor have the same anatomical plan, this is what you call the pentadactyl limbs. Penta means 5, dactyl means dict. It comes from the word digits. So if you look at your hand, you'll find out that you have 5 digits. 1, 2, 3, 4, 5. Your legs or your feet, they are 5. They are 5 toy. Or they are, yes. So if you look at them, they are 5. So if you look at a monkey, it has 5. So if you look at this picture, you'll find out that you have human, you have 5 of them. 1, 2, 3, 4, 5. Yes. If you look at a cat, it has 5. 1, 2, 3. They have 5. If you look at a whale, yes. If you look at it outside, it is different. But if you look at its anatomy, that's why we say that we compare the anatomical plan or structure. If you look at its anatomy inside, you'll find out that it also has 5. 1, 2, 3, 4, 5. If you look at a cow, it also has 5. But some of them are fused. If you look at a chicken, it has a 5. You see them 3, 1, 2, 3 in front, another one is behind, making them 4, another one is somewhere here on top. Yes, so there are 5. So all those, it shows that at least maybe we have some line of ancestry. So it means that we shared an ancestor at a certain time t. So if we study this, it helps us to know who came from who and who diverted from who. So it shows that evolution took place. If you look at this bed, it's also, it has 5 of them if you count them. So you can compare many animals, most especially the vertebrates. Most of them, they have almost the same anatomical plan, which shows that vertebrates came from the same ancestor, but we try to divide to different areas. You go to different areas. That's why you call it divergent evolution. You start with the same ancestor, but you go to different areas, and then you modify yourself. Get an example. If it is a cat, it is modified to capture a prey. If you look at a whale, it is modified into swimming. If you look at a bat, it is modified to fly. You understand? So it means that if you look at these structures, it shows that evolution took place. You also try to do the same. If you go in water, you will find out that you won't walk the same way you walk on land. You try to imitate the fish which are found in water. If you go to air, you try to fly like a bat. So basically, that's how what you call the comparative anatomy looks like. It means that all vertebrates we share a common ancestor, but we went to different environment and then we try to modify according to where we went to. And then if you have almost the same structure, then it means that you almost share the same environment where you are staying. So another evidence is genetic evidence. When you talk about genetic evidence, you're talking about the genes. So we are saying that one of the strongest evidence for common descent come from the gene sequence. Like how? If you have the genes which are almost the same, then it means that we share a common ancestor. Or the line of descent is just recent. It means that you just diverged from each other just recently. But if you have different genes, the percentage of similarity is too small, then it means that you diverged long time ago. So you're saying that organisms are likely to have common ancestor if they have, number one, identical DNA structure. You find out that chimpanzees, they share almost 98% of DNA with humans. Then it means that we are related to chimpanzees. So similar sequence of genes, it means that the way how they are sequenced, it means that the way how they are aligned. So if you have the same sequence, then it means that we share a common ancestor. Similar portion of DNA with no functions. We have what you call the nonsense DNA. Nonsense DNA is the part of DNA which doesn't have any function. It's just there, but it can be mutated. When it is mutated, then it means that it can have a function in those organisms where that mutation has taken place. But the biggest percentage of DNA has no function. And then you're saying that similar mutations, we have some similar mutations. We will see this when you are talking about the out of Africa hypothesis, that human beings originated from East Africa. So East Africa is the mother. Then if you look at this picture shows the different genes which are in different organisms and the differences. This shows the differences, the percentage in differences of the gene. Humans with another human, a human with another human, they have 0% difference in the genes in terms of the hemoglobin. And then humans with chimpanzee, they have 1% difference. Then it means that, oh, humans and chimpanzee, we are almost the same. And then humans and monkeys, they have 8%. It means that, yes, we are almost the same, but the monkeys are a little bit different from humans compared to the chimpanzee. And then rats, we have 27%. It means that one day, we and rats were the same organisms, but we diverged long time, and then it created 27%. What about humans and pigs? I've not talked about them. And then they have a very big similarities. So it means that you guys, when you're eating, make sure that you eat what is supposed to be eaten. No, don't eat your brothers and sisters. Okay, we are saying that you have frogs. Frogs, we are two different, 67%. Chicken, two different, 45 is still big. You can eat, it's fine. But chimpanzee, you can't eat chimpanzee. You're eating, this is a real brother. So, but if you're Chinese, they told me that in Chinese, gorillas, or all those things, they eat. So basically, that is genetic evidence. It shows that if you have the similarities, then it means that you have lines of descent. Is it clear? By the way, did you know that humans, let me tell you something, this is a little bit different. Did you know that humans and the monkeys, they're almost the same people, the same organism? Yes, you share 8% difference. No, 92% similarities and 8% difference. Did you know that humans have blood group D, which you call the rhesus blood group? That's why they say that your blood group A positive, blood group B positive, or blood group B negative. Means that if your blood group A positive, I don't mean HIV, no, I mean blood group A positive, you have a rhesus blood group, which called blood group D, which was discovered in a rhesus monkey. That's why it's called. People who are not producing, like you marry someone and then you keep on having miscarriages. Okay, that's genetics. Though it's a part of genetics, but I'm not going to talk about that. But that is genetics. If we go back to genetics, you will ask about that. I'll explain how this happened and how you can solve that. But it shows that if humans have blood group D, which is rhesus blood group. And then also monkey has it means that they have the blood group of the monkeys called the rhesus monkeys. Don't you see that we are almost the same? So those are our brothers and sisters. Another one is saying that comparative embryology. Here you are describing or we are comparing the embryo of different organisms. Yeah, you guys, if you look at, let me just bring it before even I start talking about it. Here you have different embryos. If I cover this part, you won't be able to tell me that this is human, this is a rabbit, this is a cow, this is a pig. You see that you and the pig, you're almost the same. You and the cow, you're almost the same. You and the rabbit, you're almost the same. So it means that we have a common ancestor. One scientist, American scientist, said that ontogeny recapitulates phylogeny, which means that history repeats itself. When you say ontogeny recapitulates phylogeny, in English, we say that history repeats itself. What does it mean in actual sense? What does it mean? It means that during our life development, we repeat our evolutionary trends. We repeat our evolutionary trends. So you keep on repeating what you went through during evolution. That's why we are saying that at a certain time T, you had a tail. That's why when you are developing, you have a tail. At a certain time T, you stayed in water. That's why you have gills. And gills, we use them for gills are used by organisms which are found in water. That's why when you go, you drown in water, you cannot stay there alive. Why? Because you don't have gills. The gills have been modified into lungs, but the fish will stay there. You understand? So it means that if you have a gill in your developmental stages, then it means that at one time T, you had the gills. So now we are saying that the study of the development of anatomy of organisms to suit to its adult form provide evidence for evolution as embryo formation in widely divergent groups of organisms tend to be conserved. It means that you keep on or you conserve those stages you went through during evolution. For example, the vestigial structures such as tails or gills. Vestigial means those structures which had a function before, but they are no longer there. So it shows that at a certain time T, you had the tails. So because the tail is not important to us, we call it vestigial. So it decreased, it disappeared. Vestigial structures. So the tail is a vestigial structure. Yes. Gills, we no longer having gills, they modify into lungs. In human can be found in embryos early stage of development. So because we share or we look like other organisms, chicken, tortoise, salamander, fish, it means that all these organisms had a same ancestor at a certain time T, but we evolved into different species. Another evidence is cultural evidence. This is basically linked to humans. It shows how humans used to have to use these tools. When you talk about, we will see in human evolution, we have different stages. We have the Homo habilis and then Homo erectus. When you talk about Homo habilis in English, it means that handyman, the people who used to like using tool, using fire. So this is the period. So if you find this in South Africa, then it means that those organisms or those humans were staying in South Africa. And then we can calculate how many years ago did those organisms which used those tools existed. So if you do the line of, or we link them, we can see that evolution took place. And then we see that these were humans, but they try to evolve to a more advanced organisms, which we call Homo sapiens. Basically, evolution is a change in the complexity of an organism.
you start as a simple organism into a complex organism. So let's look at evolution in detail. In this topic, we are going to look at, number one, the definition of the biological evolution. What does it mean? We are going to look at differences between the hypothesis and the theory. You need to be, at least you have a clear understanding between the hypothesis and a theory. And then another one, we are going to look at evidences of evolution. So we are going to look at different evidences which support evolution. Then we are going to look at the role of these evidences to support the evolutionary trends. And among the evidences, we are going to look at the fossil records. You did this in grade 10, if you can remember. Grade 10, you talked about the fossils in Marupeng. Those are the records we are going to talk about. And then we are supposed to go even to Marupeng to do these excavations. Unfortunately, because of Corona, so we can't do that. So we will use internet so that we can understand it better. Then we are going to look at biogeography. Here we are going to look at how species are distributed in the world. Still, this is grade 10 work. So we are going to link it so that you can understand it better. Then you are going to look at modification by descent. Sometimes you call this one comparative anatomy. Specifically, that is homologous structures, because comparative anatomy, it is divided into two. We have the analogous structures and homologous structures. But here we are going to look at homologous structures. We are going to look at them. What does it mean, so that you can understand it better. Then we are going to look at genetics in detail still. How does it support evolution? Because we are saying that all these are evidences which support evolution, that evolution took place. If you say that you came from monkeys, are you sure that you came from monkeys? Can you use genetic evidence to support your answer that you came from monkey? You don't just say. In science, you're supposed to say, and then you are supposed to give the evidence to show what you are saying. You don't just say and then you go. No, you have to support your statements you have spoken about. Then we have the embryology. Is also another evidence. This one, you're looking at different embryos of different organisms, the likeness. If they do have the likeness, then automatically it means that we have some relationship with evolution. So all these ones are going to look at them in these slides we are going to talk about. So we are going to also look at variation, basically how in this case we are going to look at the species and the population. Species in specifically what is the species, what's the population, all that we are going to look at them, and then the causes of variation. Among the causes we are going to look at meiosis. We are going to look at meiosis is divided into two, that is crossing over, and then a random arrangement of chromosomes. You're going to look at them. You're going to explain them in detail. Then you're going to look at a mutation. Many people have been asking me about mutation. We're going to also talk about some bit of mutation, but still we will have another class which will talk about mutation in detail. Then we are going to talk about random fertilization is also a part, another factor which causes variation. Then you are going to look at random mating, the way how you mate. When we talk about mating, we will see it in detail, but mating is a scientific term, which means sex or reproducing. And then we are going to look at the different types of variation, that is continuous and discontinuous variation. So basically, that's what we are going to look at today. If we have enough time, we will complete the slide. If the time doesn't allow us, it means that we will have to look at other subtopics in the next class. That is tomorrow. Make sure that you come on time. What is evolution? When we talk about evolution, we are talking about the process. It's a process. It's not just one step. No, it's a process. It means that look at when you eat. When you eat, The food does not just go to the blood just like that. When you have to prepare the food, you eat the food, it goes to the mouth, it goes to the throat, it goes to the stomach. It's the process. By the time it reaches the blood, then it means that it has passed in a number of steps. So when you talk about evolution, evolution doesn't just take place just in a single event. No, it takes place with different steps. So once you combine those steps, you form what you call a process. So processes of change, these processes result in change in the genes. The moment you change the genetic composition of an organism, it means that you have changed how the organism is going to look, how the organism is going to think, how the organism is going to react. So the moment you touch the genes, it means that you have touched the core of an individual or an organism. So the process of change, this process results in change of the genes that have transformed life on Earth from its earliest forms to vastity of diversity. So it means that if you look at grade 10, you talked about the geological time scale. Say that human beings maybe were the Earth originated 5 billion years ago. 5 billion years ago, the first species which had life originated 5 billion years ago. If it is like that, if all of us we came from the same species, how possible? Oh, why is it that we have different diversity? Look at human beings, same species, but when you are Tswana looks different from Zulu. Zulu looks different from Pedi looks different from, even the way you speak. We have the light-skinned people and then the dark-skinned people. You understand? So if you look at them, still same species, but we have different characteristics, different thinking. So what causes this? This is as a result of evolution. So you have different diversity among the organisms. So this diversity will help these organisms to survive in this earth. And also say that evolution is the change in the characteristics of species over several generations. That's why I say that processes, there must be different generations. It does not take place in just a single event. It must take place over many generations. You cannot see evolution just by that. You have to have many generations. That's why some people, when they tell them that they came from monkeys or from chimpanzees, some people say that no, it is impossible. It's because those generations, you didn't see them. You have to be with many generations so that you can testify what is really happening. And then you are saying that relies on the process of natural selection. This evolution relies, those who are best suited will survive. Those who are not best suited, they won't survive. Look at what is happening today. There is evolution which is taking place. Coronavirus. Yeah? The reason why the people are still suffering. Imagine they closed the schools, the country, everything was shut down when the people who are suffering or who are sick are just 100 people. But now you are over 100,000 people who are sick. You understand? Or who have been infected. And then the process, the country is running. Why? What they are doing is natural selection. Those who are best suited, they will continue and then they will survive. And those who are not best suited, they will be wiped out from the system. And then new species or those which are resistant will continue. That's what you call natural selection. Those who have passed the papers, they will go to another class. Those who have failed, they will remain in the class. So if you leave the nature to select who is supposed to progress and who is not supposed to progress, then we call it natural selection. So basically we say that evolution depends on natural selection. The characteristics, yes, will change over generation, but this process will be as a result of natural selection. So you need to understand the concept of evolution, that change from simple to a more complex organism. That's the meaning. Evolution, it means that change from a simple form into a more complex form of many generations. That is in layman's language. If someone asks you, what is evolution? Change from simple form to a more complex form over many generations or over a period of time. That's what you call evolution. And then we are saying that when you talk about biological evolution, here we are talking about life. When you talk about, the moment you talk about bio, you are talking about life. So how does life, or the different forms of life we have, how do they evolve? Because even other organisms like the sun, they can evolve from simple to a complex. Stars, they do evolve. So now those are not living. So we are looking at those organisms which are living. All life we have in this world, how did it exist? So how did it come up? So this is what called the biological evolution. So we are saying that all present day forms of life have descended from, it means that they came from and are related to those that lived in past. You are present because of your father. Your father is present because of the grandmothers and grandfathers or the grannies. Those grannies are present because, or they were present because of the grand-grand. So the life of descendants, the life of the past must be related to the life of present with some specific aspects. That's why now, if you look at they say that if you came from okay, let me say, I will talk about that when we go to human evolution. If you say that you came from a monkey, there must be some relationship between you and the monkey, to show that okay, yes, you came from monkeys. But some people say that if you came from monkeys, then why are monkeys still alive? Oh, why should we have monkeys up to now if we came from monkeys? We will see that if really you came from monkeys, you came from chimpanzees, you came from, or you came from somewhere else. Okay, we are saying that difference may be because of the modification which is taking place over generation. So you see that as time goes on, look, basically, if you look at when you are still in primary, the way you are thinking is not the same way you are thinking now. Now you are thinking big, but you, after 5 years, you'll be thinking bigger. And then when you are too old, you will be thinking extraordinary. You understand? So there is some modification which happens in your life. So this modification, if they can be passed to the next generation, then they contribute to evolution. Look at this. We are saying that maybe we came from this, and then we modified slowly to this. Then we and then this is Homo sapiens. You see them that okay, maybe we came from this line. This is our line of descent. That okay, we came from this, we started modifying, we modified again, and then we are here. Do you think that we are stopping here? It shows that you are going somewhere. Yes, it shows that you are going somewhere. There is a small video I did, I think in 2018 or 17 there. It was talking about evolution. You see that people are more evolving to a certain direction. When you watch it, if you look at YouTube, you check it from YouTube, you'll find it there. And then you see how people are modifying this. It was my aspect that maybe we might evolve to this direction. So we're still going because the changes are still happening in this world. So let's look at what is a hypothesis and what is a theory. When you talk about a hypothesis and a theory, you need to understand this. Sometimes people confuse these two statements. So when you talk about a hypothesis, is a tentative. You know the meaning of tentative? It means something which is not permanent. It's a tentative explanation for a phenomenal fact or a scientific inquiry that may be tested, verified, or answered by further investigation. If you look at this, a tentative, it must be, it's not permanent. It must be an explanation and it must be tested. Basically, that's what we need. It must be a tentative explanation that can be tested That is a hypothesis. For example, you can say that the president of South Africa, okay, Cyril Ramaphosa, will win the next election. Do you have some, you are just saying it. It's just a tentative. It's not yet proven. So if you go and do a research and see that, okay, we'll do that, then it's being supported, then we can say that, okay, now it means that it can be a theory. But let's look at this. You must be able to test your hypothesis. You don't just say it. You must be able to test the hypothesis. It must be possible to be proven. Your hypothesis must be able to be proven, and it should either be true or false. It can be true or false. There is no problem for it to be true. There is no problem for it to be false. So it's because it's just a statement. Cyril Ramaphosa will win the next elections. It's just a statement trying to which you can go and do there. He might win, he might lose. That's why you're saying it could be true or false. Let's look at it in on our aspect of science. Look at this. Michael or Michelle, whatsoever, observes that maple trees lose leaves in the fall. This is what this person is saying. So now the hypothesis could be what causes these leaves? When you're developing the hypothesis, what causes these leaves to fall? What causes these leaves to fall? So basically, you might say that he might then propose a possible explanation for the observation he has seen, and say that, okay, cold weather causes maple trees to lose their leaves in the fall. This, can you prove this? Can you test this? That cold weather causes the maple trees to lose their leaves in the fall. Can you test this? So if you can test it, yes, then it becomes a hypothesis. This statement could be false. It could be something else which causes these leaves to fall. It may not be the cold weather. So that's why you say that it could be true or it could be false. It could be something else totally different. Not even the weather. Not even the weather. It could be maybe water is not there. It could be a disease. It could be something different. It could be genetical, like the deciduous plants, whereby they lose their leaves during the drought season. It's not about what is, whether it's too cold or it's too hot. No, it's about, it tests and then knows that genetically, knows that, okay, and this period I have to lose my leaves. So this is the statement which can be proven yes or wrong. So a hypothesis is the statement, is just a statement which you can prove it right or wrong. Then when you talk about a theory, what does it mean? When you talk about a theory is a broad explanation for events that is widely accepted as true. A theory. So now if you have different hypotheses, so you have the different hypotheses, the theory must be, and the hypothesis is tested to be true, and has been repeated, and then it's being supported by evidences, then the hypothesis turns into a theory. So the theory must be supported by the facts. It must be supported by the experiments. It must be supported by the evidences. So it must be supported by a number of facts so that it turns from the hypothesis to a theory. Remember, I said that hypothesis is just a tentative. If you look at it, it's just a tentative explanation. Yes, it's just a tentative explanation, while the theory, we are saying that it is an explanation for the event that is widely accepted. So the other one is just tentative. But after proving it right, is true, is correct, then it becomes what you call the theory, or an explanation which is accepted. So this must be accepted widely. Just to go through. And then we are saying that to become a theory, hypothesis must be tested over and over again, and it must be supported by a great deal of evidence. It must be supported. You don't just say that I have a theory. No, you have to support the theory by testing it and accepting these aspects you have tried to test. Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. Evolution in present time. What happens? Does evolution takes place? We want to see it because we are saying present time. Then it means that evolution is taking place. Let's look at it in detail. Describe how mosquitoes develop resistance to DDT. How did it, like when you apply DDT, it does not affect mosquitoes anymore. Then it means that there is a resistance which has been what? Which has been formed. So we need to know that. So that is the evolution. It means that evolution, remember I told you that evolution is increasing the complexity of an organism. The organism become more complex. If the organism become more complex, it means that what was working on it, it's no longer working on it. It has become more complex. It has modified its structure. Then it means that it cannot what? It cannot be affected by such. So you're saying that now we have these mosquitoes, yes? If the mosquitoes, you have this kind, you have this kind, you have this kind. So what happens when you apply the first application of DDT? Yeah, you see that all these ones, they die. Yes, you see them that they have died. Only this and this, they have survived. So it means that there is variation among the species. There is variation among the species of mosquitoes. Yes, during the first application of DDT, yes, those which are resistant will survive, while those which are not resistant will die out. Those which are resistant will reproduce in large numbers and they will pass their genes to the next generation. Yes, when they pass their genes in the next generation, they reproduce in large numbers. And then when you apply again DDT, it means that it cannot affect them. So it means that now the mosquitoes have become resistant to what? To DDT. So first application, there is variation among these mosquitoes whereby some are resistant and some are non-resistant. But the one which are non-resistant are very many compared to those are resistant. So when you apply many which are non-resistant, they're going to die. You see them, they're fading out. While those which are resistant will survive. Yes, these ones which are resistant will pass their genes to the next generation, will produce in large numbers and pass their genes to the next generation. The second application, yes, of DDT, yes, they will not be able to affect them. Why? Because the one which are resistant have survived. Then it means that the DDT will become ineffective to the mosquito. It happens in when you take drugs and you don't complete the dose. It happens in HIV, it happens in bacteria. Yes, it happens in their life. So let's look at it. There's a large number of degree of variation in the mosquito. Yes, we saw it. When the DDT was first used, it killed off the large number of mosquitoes. But some mosquitoes were resistant to the DDT and they survived. Those which are resistant, the black one, you saw it, they were resistant, so they survived. When they survived, they passed those that survived were able to reproduce. They reproduce in large numbers, increasing the population of resistant mosquitoes. Yes, continued use of DDT had little effect on the resistant mosquitoes. It means that those which are resistant, now they have conquered the environment, so DDT will not be working on them. So at the end of the day, you'll find out that DDT will become ineffective to mosquitoes, or it becomes ineffective in killing the mosquitoes. Hence, we are saying that hence the resistant mosquitoes increased, and then non-resistant mosquitoes, they died or they decreased in numbers. So basically, that's it. That is the present day evolution. Other examples which can help you, you can do it with your teacher. Number one, this is resistance in bacteria. Yes, development of resistant strain of tuberculosis, that is bacteria, Mycobacterium tuberculosis, due to the mutation. So this the bacteria, it will reproduce in large numbers. As it is producing large numbers, it produces some resistant. When you apply the antibiotics, yes, these ones which are not resistant will die out, and those which you see they have died out. Those which are resistant, the two of them, they will survive, and then they will reproduce in large numbers. They will pass their genes to the next generation. After many generations, or after successful application of the antibiotics, yes, the antibiotic won't be able to work on the bacteria, and then it means that the resistant one will survive. That's why when they are giving you medication, sometimes they give you a combination of medication, because they know that there could be some resistant, so they must give you the medication in different versions, so that even the resistant ones, they can be resistant to this drug, but they might not be resistant to another drug. So they give you multiple drugs. But what other people do, they just take one, and then when they feel fine, they stop taking medication. You are just creating monsters in your body. You have to take the drug completely without leaving them, the way how the doctor has told you to do. So even if you're fine, you have to complete the dose, so that you kill this resistant and non-resistant completely out of your body. Then another example is HIV resistance to antiretrovirus, ARV, antiretroviral, and antiretroviral, that is ARV, antiretroviral medications. So you see that if this is the HIV, what happens? You have some resistant and some non-resistant, some resistant and non-resistant. When you apply the first medication, the resistant ones will survive, will keep on surviving, while the non-resistant will die. You see that the red ones are dying. And then after many generations, the non-resistant ones will die out, and then the resistance will survive until the end of the day. You pack. So it means that now that medication of HIV cannot work on you anymore. Yes, it cannot help you out. It means that you have to go back and then change the medication. That's how present day evolution is happening. Present day evolution, you can also have another one in black peppered moth, and then the white peppered moth, industrial mechanism, the black peppered moth, and then the white peppered moth. So you can have also such examples. So basically that's it. And then human evolution is switching in. Then I will see you there when you come to human evolution. Don't forget to subscribe for our new videos. Welcome back to our class. So today we are going to be looking at punctuated equilibrium and gradualism. So we are going to compare the two and see where do they differ. Don't forget to subscribe if it is your first time to visit our channel, and don't forget to like our video. So if you look at these two diagrams, one is showing a punctuated equilibrium and then another one is showing gradualism. Gradualism, it shows that a species change gradually. They change gradually over a great period of time. There is no rush in the changing. They change from this point to this point, but the change is gradual. It is not fast. It's just a gradual change. From this point, you see that this rat is trying to change to another color and then to a lighter color and then a very light color or bright color. And then if you look at this, still is the same. It shows that there is a gradual change from this point to this point. It didn't happen just in a single event. It happened over a long period of time, over many generations. That is gradualism. While in punctuated equilibrium, you see that the rat started from here. Yes, it's the same as this. So it keeps the same for a long period of time. But this rat is the same which gave rise to this. Yes, so when it reached here, suddenly, yes, within a short period of time, it changed from this color to this color. So when it was here, it changed from this to this immediately without a long period of time. So in punctuated equilibrium, it shows like maybe a mutation occurred and then the organism changes rapidly within a short period of time and then becomes like this from this point to this point immediately, and then it remains like that for a long period of time. Or when it reached here, it changed from this color and then went to this color just within a short period of time. While if you look at this, from this color to this color, it took a long period of time for it to change from this point to this point, and also took a long period of time to change from this to this. So if you compare this route and then you compare from here to here, this one shows that it's just a very short period of time. It happened, it changed within a very short period of time, while this one took a long period of time. The same story here, it shows that this rat just changed within a short period of time. It became like this. If you compare it with this, so it took a long period of time. So gradualism, in actual sense, gradualism require a long period of time for it to happen, while punctuated equilibrium requires a very short period of time for it to happen. And then gradualism, it is gradual, while this one, it has some periods of no change and then periods of rapid change. Periods of no change, if you look at it, and then periods of rapid change. So it has a period of no change and then has a period of rapid change. So that is what we call gradualism and punctuated equilibrium. Let's look at it. Saying that it explains this punctuated equilibrium, it explains the speed at which evolution takes place. At which speed is evolution taking place? So we are saying that evolution involves long periods of time, where the species does not change or change gradually through natural selection. This is known as equilibrium. So if you look at it, it is from this point to this point. It's the same period of no change. It is just the way it is. Or we can say a period of it's just gradually through natural selection. Gradually, this is the gradually through natural selection. Period of no change, they're talking about this. We are talking from this point to this point, from this point to this point, because these rats are the same. They are the same. They are not changing. But if you look at gradually through natural selection, then this is the period you are talking about, or this is the period you are talking about. So during this period, yes, we call it equilibrium. And then we are saying that this alternates. It has period of no change. It alternates. This alternates with what you call punctuated. That is short periods of time, where rapid changes occur. This is through natural selection, during which new species may form in a short period of what? In a short period of time. So we are seeing that from this point to this point, rapid change happens. Yes, it could be also as a result of what? Natural selection, but species form within a short period of what? Within a short period of time. Basically, that is what we call punctuated equilibrium and gradualism. Gradualism, you find out that organisms, they take a long period of time before they change, while punctuated equilibrium takes a short period of time. There is a long period of no change, long period of no change, and then rapid change, long period of no change or rapid change. They can ask you a question. Explain how did the tiger came up with stripes using gradualism or using punctuated equilibrium. So in this case, for when you explain in terms of punctuated equilibrium, you have to bring the there was a rapid change. You have to include the spots became bigger and then became stripes within a short period of time. So there was just a short period of time, then these tigers with spot became stripes. And then for gradualism, you have to tell us a long period of time. It took a long period of time before these tigers came up with the stripes. Let's look at artificial selection. What is, because we have Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. Let's look at artificial selection. What is, because we have talked about natural selection, nature selects which one is best suited to the environment. But here, artificial selection, it's the human which selects which one is supposed to continue with life or not. Which characters do you need? Actually, it depends on the desire of the human being. It's where humans select for a desirable characteristic or select for desirable traits in agricultural products, animals, rather than leaving the species to evolve naturally or gradually without human interference. So in this case, we have humans being interfering what is happening in the what? In the environment. So humans select, maybe I want birds with or chicken with big eggs, which can produce big eggs. So I'll be selecting them. For a long time, humans have been using this kind of method during breeding experiment to develop organisms with a selected set of desirable characteristics. So they keep on selecting those which are having desirable characteristics, then they select them and then they keep them. And those which don't have desirable characteristics, they remove them from the what? From the system. An example, you can find that they do select because of the following reasons. Number one, increase the quality and the quantity of milk produced by cows. Sometimes you can ask you the functions of artificial selection, the benefits of artificial selection. So these are the things you can give us that results in increase the quality and the quantity of milk production or produced by the cows. Then also drought-resistant organisms, they select for those organisms which can resist the droughts. And then disease-resistant organisms also select, they want organisms which can resist diseases. Yes, so these are benefits also of artificial selection. And then increase sugar content in the sugarcane, because they want to produce a small sugarcane can produce a large amount of sugar. So there are some of the examples. They study with these dogs. You find out that they want these characteristics. So these dogs which have such characteristics, they are being kept, they are being bred, so that after so many generations, you end up having such. The same story here, they started with this. You see 1957, the chicken was like this. 1978, it was like this. 2005, it's like this. So it means that the one which can have big meat or can produce meat, then they are being what? Selected for. And then those ones which can produce big meat or large meat or high quality meat, then they remove them from the system. The same story here to the maize and the wheat. So this is the process. What happens? Suppose you wanted a variety of cow that can produce a lot of milk. So what happens? You choose or select the cow in your herd that produce most milk. So you are looking for the cow which has the characteristic you are looking for, or a character you are looking for. And then you let these cows reproduce. You let them mate, and then they reproduce. Then they will produce kids. They select select the offsprings that produce the most milk. So the offspring will be produced, and these offsprings will be having those which can produce more milk and those which cannot produce more milk. So those which can produce more milk, you keep them. So you keep on selecting them until the genes become pure. Then you're saying that you let only these offsprings to reproduce. The one which can produce milk, you let them reproduce. Keep repeating the process of selection and breeding until you achieve your goal, until those animals which can produce milk are obtained. So what are some of the differences between natural selection and artificial selection? So we have seen that in natural selection, environment, yes, it occurs in environment. So the environment or nature is the selective force, while in artificial selection, humans select. So human is the selective force. Here in the environment select which one is the best suit, while this one is human. Like desirable characteristics. And then number two, selection is in response to what suits to the environment. Which character can survive in the environment, while here it satisfies the human needs. It's not about which character is suitable in environment, no, what sustains the human needs. And then this one occurs within the species, while this one's it can occur in one species more than one, yes, because you can crossbreed from between two species, yes, but while this one occurs in one species, strictly in one species, yes. So that's some of the differences between artificial selection and natural selection. Let's look at speciation. Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. What is speciation? Speciation is the formation of new species. So what is a species? We saw it time before. We saw it that a species is the group of organisms which can interbreed to produce a fertile offspring, like which process leads to the formation of this new species. So let's look at the process which results in the formation of new species. So here you have a population. This population is population 1 and population 2. These two populations, they can interbreed. They have the same kind of species, but now the geographical barrier has come in. So we are saying that a population of a particular species may be split into two by a geographical barrier. So this was, for example, it was one population. Actually, it was one population. There is gene flow. The gene flow means that they can interbreed, they can have sex, they can meet. Yes, for example, population of humans in South Africa and the population of humans in America. Though the distance is great, but you have planes which can go there. You have sheep which can go there. Still, we can interbreed to produce what? Though we different populations, but still we have the same species. Why? Gene flow is still there. So the population can be separated. We are saying that can be split into two by geographical barrier. Geographical barrier could be a river, mountain. It could be even a box, depending on which kind of species are you talking about. It must be a barrier which is separating them. If it is in the environment, then it could be environmental barrier. If it is in a lab, like bacteria, it can be just a small box. Yes, so it depends on how big is the species. So now we are saying that as a result, two parts of population cannot interbreed. So now a geographical barrier has been introduced here. Yes, and then now these two, they cannot interbreed. There is no gene flow. There's no gene flow. This one cannot have sex with this. Why? Because of the geographical barrier. Therefore, there is no gene flow between the two. It means that these ones cannot cross this side and these ones cannot cross this side. Yes, And then you're saying that natural selection occurs independently. So because of the different environmental conditions this side and different environmental conditions this side, then natural selection will occur differently because of the difference in the conditions of the environment. So you're saying that this is due to different environmental conditions. Yes, it means that natural selection will occur differently because of the different environmental conditions. The different environmental conditions will result in natural selection occurring independently. And then you're saying that as a result of two populations become, as a result, the two populations become genotypically and phenotypically different over a period of time. So as many generations pass, due to this geographical barrier, they cannot interbreed. They start to change, and then these ones will be wiped out because they're not being favored by the environment. The red one will survive. Also, these ones will be wiped out. The blue one will survive, and after so many generations, you'll find out that they will become genetically and phenotypically. Genetically, in terms of gene, and phenotypically, the way you see them, different colors now. Yet they were the same color, they become different over many generations. Because of the genes have changed, then automatically even the phenotype must change. And then if you remove the barrier, we are saying that even if the population mix at a later time, yes, if you remove the barrier and leave this population to mix, yes, what will happen? They will not be able to interbreed again to produce a fertile offspring. They won't be able to interbreed to produce fertile offspring. Some of them they can interbreed, but they may not produce a fertile offspring. We will see this one as we come in. So what does it mean? It means that one or both of the population have become new species. In this case, the two populations have become new species. Why? If you look at the original kind of species, is different from what is being formed. So that's how you can explain speciation. But when you are bringing in the exam, we might not bring something like this. We can bring it like this, or we can bring something different. We have given you an example. We give you an example whereby you're supposed to apply what you learned. Yes. So if the example is talking about elephants, you must explain based on elephants. If you're talking about maybe bacteria, you must explain based on bacteria. Birds, any kind of organism they are brought, as long as you talk about geographical barrier, you have to tell us which geographical barrier is there. Two, they cannot interbreed, yes. There is no gene flow, yes. You must talk about natural selection and occurring independently, and you must talk about due to the environmental conditions. So you must talk about the genetically different, resulting in phenotypically different organisms. And then you must talk about when you remove the barrier and leave them mix, then you won't be able to interbreed to produce a fertile offspring. Then you tell us how many have become new species. Then you'll be able to get the mark. Describe how these two species of the flies were formed. So now it means that now they have brought an example of flies. How did they form? If you look at these flies, as if they're in the lab, yes, they are being separated by the what? Box. At first, it was one population, as we told you. It was one population. Now the population is separated by the box. Yes, the geographical barrier now. The barrier here is now the box. This one, there is no now gene flow. You see that they are still the same kind of species. So there is no gene flow between them. These ones cannot come here and then interbreed with this. And then because of there is no gene flow and they are being separated differently, then they have different environmental condition. In this case, what is the different environmental condition? It's the kind of food which is present. Here we have starch food, and then here we have maltose. So it means that they have different environmental conditions. So those which cannot feed on starch, they will do what? They will die. And those which can feed on starch, they will survive. While here, those which cannot feed on maltose will die, while those ones which can feed on maltose, they will survive. Yes. So over many generations, yes, it means that due to no gene flow between the two populations, yes, then this will result in a different genotype, this will lead to these species becoming genetically different and phenotypically different due to the different environmental conditions as a result of natural selection occurring independently. So the different genotype resulted in different what? Phenotype. And if you remove the barrier and you leave them mate, they cannot interbreed to produce a fertile offspring. So it means that now this and this, they have become new species. So that's how you can explain, depending on the question they have given you. You have to be keen. You have to look at which barrier, which kind of environmental condition, which kind of species have been given. So, and then how many have become new species. If you look at them, yes, you'll find out that they are different species. If you look at this, is different from this, and this is different from this, then it means that the two have become different species. There was one population of flies. The population of fly species became separated by the wall of the box into population A and B. Yes, we saw it. And then because there is no gene flow between the two populations separated, yes, what happened? Natural selection occurs independently in each A and B due to exposure to different environmental conditions. In this case, that is the food source. The food source. The food was different. So because the food source was different, this one fed on maltose, and then the other one fed on what? On starch. So that the environmental conditions were different. Hence, this will result in the population becoming very different from others. That is genotypically and then phenotypically. You have to start with the genotypically and then talk about phenotypically, because it's the genotype which determines, the phenotype. And then you're saying that when the population were allowed to meet again, they were not able to interbreed. So when you remove the box and then make them mix, they are not able to interbreed to produce a fertile offspring. Don't forget this. And then you're saying that the population A and B are now new species. So they have become new species. Yes. So P means a point or a mark. It could be. Yeah. So it should not confuse you. So basically, that's it. And then you're saying that another question is this. You can also apply it in this case. Say geographical barrier, they have not talked about it. You see that it was one population divided. You see now it starts to change, then finally changes, and then it forms different two new species. So it's almost the same as the previous one, but now it is in a different angle. Yes. Try to download these slides, and then you can try to practice such questions. Yes. So what are some of the mechanisms of reproductive isolation? When you talk about reproductive, it means that sex is not happening. Yes, it has been isolated. Organisms have been isolated from sex. That is reproductive. They are not reproducing. It results in them not reproducing. So that isolation, what are some of the mechanisms? What are some of the things which result in these organisms not reproducing? Keeping species separate. So number one, breeding at different times of the year. Yes. So this one breeds in fall, and then this one breeds in late of the winter. So it means that they cannot have any time when they are breeding at the same time. So they are being separated by the different times of the year. Number two is species-specific courtship behavior. Like the way how you do your courtship is different from how dogs do the courtship, is different from how the birds are doing their courtship. So it is different. We have different ways in which we do our courtship. We seek for sex differently. We have different ways. So that one can also, the way how you do it and the way monkeys do it, it must be irritating you, and yet the monkeys are enjoying the same story to humans. So if it happens the way how we do courtship, then it means that it could lead to isolation, adaptation to different pollinating agents or pollinators. You see that here, this is an insect, this is also insect, but it is a butterfly. This is another insect, kind of different insect. have different insects. This is a bird, this is a bat, this is a human. So they have different pollinating agents or pollinators. So because of that, they are not, if this cannot come, then it means that you will not carry out pollination, or it will not reproduce. So because of that, they are being isolated, or they are being separated. Then you have, yes, you can have sex, you can have what, you can produce a baby, but you end up producing an infertile offspring. For example, a horse, which is 64, so when it is one end, when meiosis takes place, it is 32, and then a donkey, which is 62, 31, when it divides, then they produce 32 plus 31 produces what? Produces 63. So when it is going to carry out meiosis, what is going to give? It's not, it is not applicable. Then it means that this one will be, it won't be able to reproduce. So it is infertile. They have crossed, they have produced a baby, but the baby is not fertile. Yes. So it cannot reproduce again over and over and over again. So it becomes still where they are being separated. And then lastly, prevention of fertilization. So you hear that a person produced a dog. Does it mean that that person slept with the dog? No, it doesn't mean like that. It could be mutation. But now, is it that if you produce a snake, produce a dog, or produce whatsoever, is it? It's not that you slept with the dog. It's because we have different kind of sperms and ova. Yes, so fertilization will not take place. You see, the egg of a human and the egg of the bird, the chicken, they are totally different. Yes, humans are big, but their eggs are small, while birds are small, but their eggs are big. So there's no way how you can reproduce. These are some of other examples of speciation. How did they form these beaks? Here is another example which tries to, how did these two species came about? The question can come, how did they come about? All these are other examples of speciation. So you can read about them, or just have one the way I've explained it, then you'll be able to apply others. Okay, let's look at variation. So differences among organisms of the same species, we call it variation. So when we talk about variation, we are talking about this organism. What are some of the causes of variation? These causes could be genetic, or it could be environmental. You can be tall, not because you are genetically tall. It could be environmental. You can be short, not because you are genetically short. It could be environmental. Haven't you ever seen father, mother, both short, but they produce a tall kid? It could be genetical, yes, or it could be environmental. Oh, dark skin, or light skin, they produce a dark skin, and then the father said, this is not my kid. Who told you it's not your kid? It's whose kid? So it's your kid, but some environmental factors could happen, or some genetic factors could also happen. When we talk about a species, it's a group of organisms living together. And these organisms must interbreed to produce a fertile offspring. I've tried to explain this. A group of living organisms consists of similar individuals, meaning the same group, capable of interbreeding. Interbreeding is sex, having sex and produce kids. That's interbreeding. Courtship, you could do the courtship and then you produce. I hope everyone is above 18. To produce fertile. Fertile means something which can reproduce again, or viable offsprings. Offsprings are babies. We saw them when we were talking about genetics. And then a population, if you have dogs, a lot of dogs in the same area, specific time, then we say that a population of dogs, population of plants. So when we talk about a population, it's a group of organisms of the same species. They must be living together and they must be able to interbreed. So if they can interbreed, then it means that it's just a population of those organisms. reason why I've talked about the species and population, because variation happens in the species, or it happens in the population. That's why I try to do a recap of population and species. What are some of the causes of variation? Number one, crossing over. You remember we talked about crossing over. When we talk about crossing over, homologous chromosomes come together during prophase I. You must be specific, prophase I, and then the chromatids of the homologous chromosome, they cross over. When they cross over, you'll find out that they produce chromosomes with mixed genetic composition. You find out that this part came from this chromosome, while this part came from this chromosome. So this one came from this, and then this one came from this. So it means that genes have been mixed. That's why you have the father, you have the same mother, but you have different people. They appear, they don't even look alike. Sometimes you go even somewhere and they say like, hey man, you resemble someone I know there. Does it mean that your father went to that lady and then impregnated her and without knowing? No, it's because you share the same ancestor. Then it means that you still have those genes within us. So crossing over, it helps in when homologous chromosomes from form pairs during prophase I. I said prophase I is very important of meiosis. Crossover can occur, as you see it here. Crossover occurs, as I've tried to explain, leads to new combination of genetic material in new cells. So if they separate during anaphase, they will go to different cells. Then it means that these cells, they will have different genetic material compared to the parents. If parents had this, then automatically the offspring, you will have this. So it means that you will have some characters from the father, some characters from the mother, but not all characters that you will be the same, exactly like the father. So crossing over helps in bringing about variation. Number two, random arrangement of chromosomes. How do chromosomes randomly arrange? If you look at this, you'll find out that you can have this, maybe. Remember, they just do randomly. They are like chicken when they are going to mate. Okay, I'll talk about that. For example, we don't know whether this chromosome is going to be on this side or is going to be on this side. It can be like this color, and this color, or it could be like this. So we don't know which one is going to be this one, which one is going to be this side, this one is going to be this side. And remember, here we're just showing 4 of them. They are not 4 different positions, as long as they are homologous chromosomes, but they have different positions. So you end up having different combination of, for example, in this case, you're going to have this, you're going to have this. In this case, you're going to have this, you're going to have this, and then you have another combination, you have another combination. So you end up having many combinations. So random arrangement of chromosomes during metaphase result in the gametes with unique. You will find out that you have unique combinations of what? Of alleles. At the equator during metaphase allows different combinations of chromosomes or chromatids to go into each new cells, resulting in meiosis, making them different. So if you look at this sperm will be different from this sperm, and this sperm will be different from this sperm. All these, if you look at them, all of them, they are different, but it came from only 2 combinations, but you have many combinations. You have many combinations. So because of this, it will result, and you don't know which sperm is going to fertilize the ovum. So because of this, then it helps in bringing about variation. And then another one is random fertilization. Look at this egg, and look at these sperms. Look at these sperms. All these sperms, they are looking for this ovum. And we don't know which one is going to fertilize the ovum. You see, we don't know. So if it is like that, it is by chance, it's random. All these sperms are different, but we don't know which one is going to fertilize this. So if the guy produces like 100 million sperms, and then only one, and each sperm is different from the other, only one will fertilize the ovum. So what does it mean? It means that this time it will be a different sperm fertilizing. Another time it will be a different sperm fertilizing. Obviously, we will have different genetic makeup, and then we will have what you call variation. We will have some differences among us. That's why you have different colors, you have different heights, you have different sizes, you have different length of hair. You have why? Because of the genes are being mixed up during these processes. So you're saying that any sperm stand a chance of fertilizing an egg or cell. You see how fast your sperm was. I told you that you were lucky. How fast your sperm was to fertilize the ovum, so that you at least you can exist. If your sperm did not fertilize an ovum and then another sperm fertilized, you think you would have existed. Someone else would have existed, not you. But your sperm was smart, was faster, everything was perfect, and then it was able to fertilize the ovum, and then you are existing. So you're saying between different ovum and then different sperm cells formed by meiosis, resulting offsprings that are different from each other. We saw that you have different sperms. Each sperm or each cell is different from the other. So it shows that random fertilization can help in bringing about variation. Note, each individual is capable of producing over 8 million sperm. You see? Hey, do you know the meaning of 8 million sperm? If they give you 8 million rand, are you going to be rich or poor? Obviously, you're going to be rich. So now imagine that is sperm. Yeah, 8 million sperm are potential. When you talk about potential, it means that those which can fertilize, because we have other sperms which cannot fertilize. But random chance of any one sperm and egg coming together is a product of these two possibilities. Look, 70 trillion different combinations. That is the chance. You see how it means that you won a barrel of 70 trillion. 70 trillion. Even the budget of South Africa might not be 70 trillion. I don't know. I'm not a politician, but it